Hello, my dear students. I hope you all are doing great. My name is Biswajit. I'm your botany teacher. Today, we are going to discuss a very important chapter of class 11, and the chapter is cell cycle and cell division. And in this class, we will talk about what is cell cycle, what are the different phases of cell cycle, what is mitosis, mechanism of mitosis, significance of mitosis, then what is meiosis, mechanism of meiosis, and significance of meiosis. So these are the things which we are going to cover in this lecture. And because this chapter is very conceptual, so I advise you to watch this lecture with full concentration. So the first thing which the NCERT says is that growth and reproduction, they are the characteristics of what? Organisms as well as the cells. So what do you understand by growth? This is a feature which is observed in both living organisms as well as non-living entities. And all the living organisms, whether they are prokaryotic or eukaryotic, unicellular or multicellular, all organisms, they show growth. And the growth in organism is always internal. And in organisms, the growth is irreversible process in which there is increase in number of cells and mass. And if I say cell shows growth, that means there is increase in the size of cell, there is increase in the mass of cell. And how does this take place? Due to increase in cytoplasmic content and nuclear content. Cytoplasmic content increases, that means there will be increase in the number of cytoplasmic organelles like mitochondria, chloroplast, endoplasmic reticulum, Golgi body, etc. And there will also be synthesis of different chemicals in the cytoplasm like nucleotides, amino acids, proteins, etc. Okay, and what about the increase in nuclear content? This happens due to DNA replication and also due to increase in the amount of histone proteins in the nucleus to fold the DNA molecule, to package the replicated DNA molecules. Okay, and when I talk about reproduction, this is a feature which is observed only by living organisms, and this is not observed by all living organisms. There are certain organisms which do not reproduce. Okay, and if I talk about reproduction with respect to cell, so what do you understand by cell reproduction? See, when cell divides, new cells are formed. That means cell is capable of forming new cells. That means I can say cell can perform reproduction. I did not say cell will always perform reproduction. Why? Because there are certain cells which have lost their ability to divide. In case of plant anatomy, you might have heard about permanent tissues, okay, like sclerenchyma. They will never divide. Collenchyma, they can divide. They can regain the ability to divide. Parenchyma, they can regain the ability to divide. Okay, so cells, because they divide to produce the new cells, so I can say cells have the ability to reproduce. And when I talk about the reproduction in organism, depending upon the involvement of sex, that is the process of fertilization, the reproduction can be of two types, sexual and asexual. Okay, and whenever there is sexual reproduction, there will be sex. Sex means fertilization. In fertilization, there will be fusion of male gamete and female gamete. When they fuse with each other, they will produce zygote. And every organism which reproduce sexually, they start their life from a single cell, that is zygote. So suppose this is a zygote, and zygote is a cell, that is a fertilized egg, that is a cell. So this zygote will develop into an adult body. So suppose this is you, this is your adult body. Okay, zygote consists of only one cell, whereas adult body consists of many cells. So during this development, what is happening? There is increase in number of cells. There is increase in a number of cells. Okay, so I will say to increase the number of cells, the process which will take place is cell division. Cell division, and multiple rounds of cell division will take place. Multiple rounds of cell division will take place to produce many cells. Okay, and the cells which are present in the adult body, they are of different types. So to produce different types of cells, the process which will take place is cell differentiation. So I can say that the development of an adult body from the zygote is achieved by multiple rounds of cell division and cell differentiation. Okay, and let me tell you one example of what? Cell growth. Let me tell you one example of cell growth. In the chapter of plant growth and development, the watermelon cell, the example of cell growth is given. The watermelon cell can increase its size up to 3,50,000 times. How many times? 3,50,000 times its cell can show increase in size. Okay. Now, so let's talk something about cell division. What? Cell division. So what do you understand by cell division? Cell division is a process in which new cells are produced from a pre-existing cell. Okay, and because due to cell division there is increase in number of cell, that is why we call cell division as cell multiplication. And because in cell division new cells are formed, that is why I will call cell division as cell reproduction. I repeat, because of cell division there is increase in number of cells, because there is increase in number of cells, so I call cell division as cell multiplication. And because in cell division new cells are formed, so I will call cell division as cell reproduction. Okay. Now, in cell division, during a cell division, a cell will perform a number of events. Okay, and those events are of very and the main events during cell division which take place in a cell are DNA replication and cell growth. And during cell growth, or that is during the growth of a cell, both nucleus will grow in size and cytoplasm will also grow in size. But if I talk about the growth with respect to cytoplasm, then we will find that the growth of cytoplasm will take place throughout the cell cycle. That means in all phases of cell cycle. And that is why I call cell growth with respect to cytoplasmic growth as a continuous process. But if I talk about DNA replication, it takes place in a specific phase of cell cycle. And what is that phase? That is S phase. This is very, very important. In which phase of cell cycle DNA replication takes place? It is the S phase. Which phase? S phase. And because this takes place in only one phase of cell cycle, that is why we call DNA replication as discontinuous. And this DNA replication can also be called as genome duplication. Why? Because of DNA replication there will be doubling of the amount of DNA. Okay, or I can say doubling of the amount of genome. You may ask me, sir, what is genome? Genome is basically sum total of DNA present in a gamete or haploid set of chromosomes. Okay, with respect to human being, a genome will consist of DNA of chromosome 1, chromosome 2, one copy of each chromosome. Okay, one copy of DNA of each chromosome, DNA 1, DNA 2, DNA 3, like this, 21, 22 and X or Y. Okay, and because due to DNA replication there is doubling of the DNA or doubling of the genome, so I call DNA replication as genome duplication. Okay, and whenever there is a cell division, both cell growth and DNA replication takes place, and all these events they occur in a highly coordinated way. And because of this highly coordinated DNA replication and cell growth during cell division, the cell division is proper, and the daughter cells, DC's daughter cells which are formed are of proper type. That means they will have intact genome. Intact genome means they will have every chromosome. They will have every chromosome. They will not miss any chromosome. Okay, I repeat, during cell division, both cell growth and DNA replication they occur in a coordinated manner. So what is the result of this coordinated happening of or occurring of cell growth and DNA replication during cell division? I will say the cell division will be proper cell division, and the daughter cells which will be formed, they will have intact genome. Intact genome means the genome which has every chromosome. No chromosome will be missed. Okay, now what about cell cycle? And the cell cycle we are going to talk with respect to eukaryotic cell cycle. Okay, so to explain that, let me tell you with the help of diagram. So suppose this is a cell, and the cell which undergoes division, that is called as parent cell. So this is parent cell. This is parent cell. Okay, and what happened? Cell division took place. Here cell division is taking place, and in cell division, in cell cycle, the phase in which division actually takes place is called as mitotic phase. Which phase? Mitotic phase. Okay, so this is mitotic phase, and here cell division is taking place. Okay, which phase is this one? This is mitotic phase. So let me write here, this is mitotic phase. Which phase? Mitotic phase. Okay, so this is basically mitotic phase. Okay, mitotic phase. So after cell division, I will get daughter cells. So these cells are daughter cells. These cells are daughter cells, and you can clearly see that the size of daughter cell is small as compared to the smaller as compared to the parent cell. Now, before dividing, before undergoing division, this daughter cell has to grow. It has to grow. Okay, so what is happening over here? Here growth is taking place. What is taking place? Growth is taking place. Where? In the daughter cells. So after growing, these cells will be okay. These cells which are formed just after the division, they are called as immature daughter cells. They are called as immature daughter cells. And after growth, these daughter cells will become mature. They will become mature daughter cells. Okay, and these mature daughter cells, they will now act as parent cell. They will now act as parent cell, and this parent cell, they will undergo cell division. They will undergo cell division. Okay, and when there is division, that means which phase is taking place? Mitotic phase is taking place. Which phase? Mitotic phase is taking place. And the phase between two mitotic phase, this is one mitotic phase, this is another mitotic phase. Okay, both see, both these cells, they are undergoing mitotic phase simultaneously. So I will say this is one mitotic phase and this is another mitotic phase. So the phase between two mitotic phase in which growth takes place is called as interphase. So this is basically interphase. Which phase? Interphase. So this is interphase. Okay, so the cell is undergoing through passing through interphase. Okay, and in this interphase, thus various subphases are what are the subphases of in this interphase? G1 phase, S phase, and G2 phase. Okay, G1 phase, S phase, and G2 phase. And we know in the S phase of cell cycle, what happens? DNA replication will take place. What will happen? DNA replication will take place. In this S phase, what is going to happen? DNA replication will take place. Which one will take place? DNA replication will take place. Okay, and see, these daughter cells, they will again undergo growth and they will finally become mature daughter cells, and they will act as parent cell and they will divide to form new daughter cells. So these things, that means growth, division, they keep on happening. They keep on occurring. So I can say mitotic phase and growth phase, that is interphase, they occur in a cyclic manner, and all those things, they are collectively called as cell cycle. Cell cycle. That means I will say that the events which take place in a sequential manner, which includes interphase and mitotic phase, or I can say the events which take place from one cell division to the next cell division, that is called as cell cycle. Okay, this is one cell division, this is one cell division. So after one cell division, what is happening? Growth is taking place. In which phase? Interphase. And then what is happening? Cell division is taking place. Mitotic phase is taking place. Okay, and this interphase and mitotic phase, they are collectively called as what? Collectively called as cell cycle. Or I can say series of events which take place from after one cell division to the next cell division, that is called as cell cycle. Okay, and in this case, so you can see sequence of events from one cell division to the next, or sequence of events by which cell duplicates its genome. Okay, cell duplicates its genome means there is DNA replication. Okay, you can see in the interphase what is happening. DNA replication is taking place. Okay, and increases cellular component. Why? Because in interphase there will be increase in this cytoplasmic organelle also. Like there will be increase in number of endoplasmic reticulum, Golgi body, okay, mitochondria, plastids, etc. Okay, so in the interphase there is increase in what? DNA content, as well as increase in cytoplasmic organelles, and eventually it is undergoing division. So all those events which occur in a sequential manner, that is called as cell cycle. Okay. Now, what about the uh, other features of cell cycle? See, the events which are taking place in during cell cycle, they are ultimately controlled by genes. So I will say cell cycle is genetically controlled, and to in order to take in see and the process of cell cycle, or I can say the events which take place during cell cycle, they require energy, they require ATP. Without ATP, without energy, without metabolic energy, those events will not take place. Okay, and that is why I say this process of cell cycle is energy dependent, and this process is irreversible. This is irreversible. Okay, let me explain you what is irreversible. Okay, see, suppose this is the cell cycle, this is not a cell, this is the cell cycle. Okay, and this phase is G1 phase, this is S phase, this is G2 phase. Okay, this is prophase, this is metaphase, this is anaphase, this is telophase, and this is cytokinesis. All these phases collectively called as, they are collectively called as cell cycle. Suppose the cell has completed G1 phase and it has entered S phase. It will never come back to G1 phase. If the cell enters into S phase, it will never come back to the G1 phase. That means it is irreversible. Okay, or suppose the cell has just completed the S phase and it has entered into G2 phase. It will never come back to the S phase. Okay, that means cell cycle is irreversible. And what is the direction of cell cycle? You have to start with the G1 phase and you have to end with the cytokinesis. And in what direction? In the clockwise manner. In clockwise manner you have to move. Okay, so suppose this is the parent cell. Suppose this is the parent cell, and this parent cell is undergoing cell cycle, and after cell cycle it will produce two daughter cells. It will produce two daughter cells. Okay, and these phases, prophase, metaphase, anaphase, telophase, they are collectively called as mitotic phase. Sorry, they are collectively called as karyokinesis, and this is cytokinesis. And all these phases, they are collectively called as mitotic phase. They are collectively called as mitotic phase. Okay, so this process of cell cycle is irreversible. This is genetically controlled, that means controlled by genes. Okay, and they require energy to occur, and they are irreversible. And the events which are taking place, no doubt they are controlled by genes, but how the genes are controlling? The genes will produce certain chemicals. Okay, and those chemicals will control the cell cycle. And what are those chemicals? One is cyclin, which is a protein, and second is the Cyclin-Dependent Kinase, CDK, Cyclin-Dependent Kinase. Okay, and we will talk about cyclin and Cyclin-Dependent Kinase in detail later on. And what about the duration of cell cycle? See, the duration of cell cycle, what will be the duration of cell cycle? That will depend upon two things. One is the nature of cell, what type of cell you are talking about. That means I can say this, the duration of cell cycle will be cell specific. Okay, that means suppose there is an individual, it has different types of cells, and different types of cells, they undergo cell division. So suppose cell A divides in X time and cell B divides in Y time. So you can say that different cells of same body, same individual, they require different times to divide. Okay, and it also depends on the species. That means this the cell of a human body, that takes certain time to divide, and the cell of elephant, that takes some different time to divide. Okay, so I can say the duration of cell division or cell cycle is species specific. That means it varies from species to species, and it varies from cell to cell. That means it is dependent on cell type, cell specific. Okay, some of the examples of species specific duration of what cell cycle. If I take about the example of what yeast takes, an yeast cell takes about 90 minutes to divide. So what is the duration of cell cycle in case of yeast? It is 90 minutes. Okay, E. coli is a prokaryote and it requires about 20 minutes to divide. Okay, and what about human cell? If I take a human cell in the culture medium, then it will require about 24 hours. It will require about 24 hours to divide. Okay, different types of human cells in culture medium, they require different duration, different time period to divide. I have taken the average over here, so the average value is 24 hours. The average value is 24 hours. Okay, so you can see in different species the duration of cell cycle is different. The duration of cell cycle is different. Okay, now what about cyclin and CDK then? See, cyclin is a protein and CDK is basically cyclin dependent kinase. We know during cell cycle different events are going to take place, and those events are performed by different proteins, and those proteins can be called as target proteins. I repeat, during cell division different activities or events are going to take place, and those events are performed by whom? By target proteins, and they will perform, so I can write here, these target proteins, they perform functions or their role during cell cycle. Okay, and when these proteins will perform their cell cycle if they are active. So the target proteins to perform their functions related to cell cycle, they have to be active. So they need to be active. So these proteins, they need to be active. But these proteins, when these target proteins, when they are synthesized, they are not in active form. So they will be activated. So suppose initially when this target protein was synthesized, it was inactive. So I have to activate this target protein. So what am I doing? I'm activating this target protein. Okay, and who is going to activate the target protein? It is the CDK, cyclin dependent kinase. So I can write here, the cyclin dependent kinase, it activates it activates the target protein. How will it activate the target protein? See, the name is cyclin-dependent kinase. And any enzyme whose name is kinase, then you can say that enzyme will perform phosphorylation. That means it will add the phosphate. So suppose this is the target protein. And no phosphate is added to this target protein. So when there is no phosphate, this is inactive. But later on, when the phosphate groups are attached, to this target protein, then it will become activated. Or I will say the target protein is now active. We know the protein will have different types of amino acids. So do you think that every amino acid in the protein will be phosphorylated? No. Certain amino acids will be phosphorylated, like serine or threonine. Which one? Serine or threonine. Okay, so I will write here serine and threonine. These amino acids of the protein, they will be added with the phosphate. Okay, and when they are added with the phosphate, the protein will be phosphorylated, and once the protein is phosphorylated, it will become active. Okay, so let me draw the diagram over here so that you can understand it more clearly. Okay, so suppose this is the target protein. Which is inactive, and suppose this is the active target protein. And when will it become active? If it is phosphorylated. P is phosphate group over here. Clear? So this is the kinase enzyme, and this kinase enzyme will phosphorylate this target protein to make it active. Okay, and this CDK, cyclin dependent kinase, this enzyme kinase will activate only if it is active. So to activate, it has to be active. If the CDK has to activate the target protein, it should be in active form. But when the CDK is formed, it is formed in the form of inactive. It is formed in inactive state. So this CDK has to be activated. Okay. So those students who are preparing for KVPY, they should remember one thing, that whenever you phosphorylate a protein, which amino acid residues of the protein they will get phosphorylated? It is serine and threonine. Which one? Serine and threonine, they will get phosphorylated. This is for KVPY students. This is for KVPY students. Okay. This is for KVPY students. Now see, this cyclin-dependent kinase, it is, when it is formed, it is inactive. It has to be activated. So who is going to activate this one? It is cyclin. It is cyclin. So cyclin will activate. Cyclin will activate which one? Cyclin-dependent kinase. So whether the CDK will be active or inactive, it depends on the presence or absence of cyclin, and that is why this kinase enzyme is called as cyclin-dependent kinase. If cyclin is present, then it will be activated. So if it is absent, it will be inactive. So that is why I call this enzyme as cyclin-dependent kinase. And remember one thing, the different activities during cell cycle, they are controlled by different cyclin and different kinases, cyclin-dependent kinases. Or I can say, during different phases of cell cycle, different cyclin and different CDK, they will operate. Okay, so you can write that point. I repeat that for you. You can note that during different phases of cell cycle, different cyclins and different CDK, they operate. They play their role. Now, what about cyclin and CDK then? See, during different phases of cell cycle, the amount of cyclin and the what about the amount of cyclin and what about the activity of cyclin? Okay, now let me tell you, if I talk about the amount of cyclin, what amount of cyclin, then suppose there is a phase, G1 phase. So during G1 phase, only G1 cyclin will be present. During S phase, only S cyclin will be present. What am I saying? Is it clear to you? See, the cyclin which play their role in G1 phase, they are G1 cyclin. Cyclins which play their role during S phase, they are S cyclin. Okay. So during G1 phase, the cell will have only G1 cyclin, not G2 cyclin, not S cyclin. And during S phase, the cell will have only the S cyclin, not G1 cyclin nor G2 cyclin. Okay, that means the nature of the cyclin or the amount of cyclin, it varies from phases to phases. It varies from phases to phases. So I will say that what about the amount of cyclin? I will say this varies with phases. It varies with phases of cell cycle. Okay, what about the activity? They are always in active form. The cyclins are always in active form. So I will say they are always active. That means I will say their activity will not vary. The activity of cyclin will not vary. The amount will vary, but the activity of cyclin will not vary. Whenever they are formed, they are always in active form. What about the examples of cyclin then? The examples I can say G1 cyclin, okay, or I can say S cyclin, G2 cyclin, okay, M cyclin, etc. So these are the different cyclins. What about CDK? The amount of CDK is always fixed. That means the types of CDK and the amount of CDK in G1 phase is identical to that of M phase or S phase or G2 phase. That means the same type of and same amount of CDK, they are going to be present. So if all the CDK, they are present in all the phases of cell cycle, then the CDK whose activity are not required in a certain phase, they have to be in inactive state, and those CDK whose activity are required, they have to be in active form. Okay, so I will say one thing, that I can say one thing over here, that the amount is not variable. The amount does not vary. The amount does not vary, but their activity varies. The activity of the CDK that varies with phases. For example, suppose there is a CDK. Suppose there is a CDK X, and suppose this CDK performs this activity in G1 phase. So in G1 phase, this CDKX will be in active form. But in S phase, in G2 phase, in M phase, that will be in inactive form. Clear? Now, what about the example? The example is CDC2. CDC2. CDC is cell division cycle. Cell division cycle. Okay. So I can say that the cell cycle is controlled by genes, and those genes will produce cyclin and CDK, and they are ultimately going to perform the different activities which are happening during the cell cycle. Okay. Now, if you are able to understand, then please comment, drop your message in the comment box, and you please tell me whether you are able to understand or not. Okay. Now let's talk about what the phases of cell cycle, and which cell cycle? Eukaryotic cell cycle. Which cell cycle? Eukaryotic cell cycle. So all the events in the cell cycle, they can be categorized or grouped in two phases. What are those? One is the interphase, and second is the mitotic phase. Second one is the mitotic phase. Okay. So what can you call this interphase? We can call this interphase as resting phase. Why resting phase? You can write this point, that because in interphase no division in the cell is taking place. See, in the cell, during cell cycle or cell division, division of cytoplasm and division of nucleus takes place. I repeat, during cell division, division of nucleus takes place, which is known as karyokinesis, and division of cytoplasm, which is known as cytokinesis, they take place. And because in interphase neither nuclear division nor cytoplasmic division are taking place, that is why this phase is called as what? Resting phase. Resting phase. Okay. It does not mean that no event is taking place. Events are taking place, but no event related to division of nucleus and cytoplasm are taking place. That is why this is resting phase. And I can also call this as non-dividing phase. I can also call this as non-dividing phase. But in M phase, there is division of nucleus and there is division of cytoplasm, and that is why you call this phase as dividing phase. What do you call this phase? Dividing phase. Okay, so this is dividing phase, and this phase is also called as most dramatic phase. This is also called as most dramatic phase. Why do you call this as most dramatic phase? You can write that point. Okay, because in M phase, during M phase, the cell will show reorganization of almost all the cell organelles. There will be reorganization. How they are going to take place, we will discuss during discussing the mitosis. Okay, I will tell you how the nuclear membrane will disappear. Okay, and they will again reappear. How the Golgi body endoplasmic reticulum, they disappear in the prophase, and how they reappear in the in the telophase, and how the chromosomes get separated. So during M phase, a number of reorganizations in the reorganization of a number of cell organelles takes place, and that is why we call this as most dramatic phase. Okay, now what about the metabolism? See, if a cell is dividing, that means that cell is living, and for a cell to be dividing, it has to perform metabolism. Okay, so do you think that during all the various phases of cell cycle, the rate of metabolism is same? No. In the interphase, the cell will be having more metabolism. That means it will be metabolically more active. Whereas if a cell enters into the M phase, the metabolism will decrease. It will decrease. So I will say in the interphase, the rate of metabolism is what? Rate of metabolism is high. or I can say the cell in the interphase is metabolically highly active, but in the M phase it is the rate of metabolism is less, or I can say low, metabolically less active. Okay, now what about the growth? As I said just before some time, I had told you that the cell growth, the growth of the cell takes place throughout all the phases of cell cycle, all the phases of cell cycle. That means it will take place in both M phase as well as interphase, M phase as well as interphase. So in which phase there is more growth of the cell? It is the interphase. It is the interphase. Okay, and that is why I will say that the growth in the interphase is very high. That means I can say the growth will be intense growth. The growth will be intense growth. And in M phase also growth will continue, but as compared to interphase, the growth is relatively less. So I will say relatively less growth will take place. Relatively less growth will take place. What about DNA replication? As I said, DNA replication takes place only in the S phase, and S phase comes under interphase. S phase comes under interphase. So DNA replication will take place in interphase, but in M phase there is no DNA replication. So what are the subphases which come under interphase? It is the G1 phase, it is S phase, and it is G2 phase. Previously as per old NCERT, G0 phase was a part of cell cycle. So you can as per old NCERT, you can also include G0 phase under the interphase. Okay, now what about M phase? In M phase it includes two subphases. One is karyokinases. Karyokinesis term is derived from two words. One is karyon means nucleus. Second is kinesis, and this kinesis means division. That means this is a phase in which there is nucleus will divide. This is nuclear division. Okay, and the second phase which comes under M phase is cytokinesis. Cyto is derived from cytoplasm. Okay, cytoplasm indicates cytoplasm, and kinesis is division. So there is division of cytoplasm. This is cytoplasmic division. This is nuclear division. Okay, so this was about M phase. Okay, what about the duration? Do you think that all the phases of the cell cycle, they will require same time? No. So I will say duration. The duration of interphase is longer. It is longer, and the duration of M phase is shorter. That means the cell requires more time to prepare itself. Okay, and this interphase is also called as preparatory phase. You can also write that point. Okay, so let me write that point over here. Not dividing phase or preparatory phase. Why do I call this as preparatory phase? Because in this phase cell prepares itself to divide. How? By doubling the number of DNA molecule, and by increasing the number of different types of cell organelles. So it is preparing itself for division of both nucleus as well as cytoplasm. So we call this as preparatory phase. Which phase? Preparatory phase. Okay, non-dividing phase and preparatory phase. Okay, here what about metabolism? The metabolism is high. Let me write that point over here. Metabolism. What about metabolism? The metabolism is high during interphase. Okay, so this was about what? Interphase and what? M phase. Okay, now let me tell you something about interphase. What? Interphase. Okay, as I have told you that the interphase consists of what? G1, S and G2 phase. Which phases? G1, G2 and S phases, right? And please look at this diagram. this is interphase, this is interphase. Okay, this is M phase, this is M phase. That means this interphase is taking place between one M phase and the other M phase, and that is why we say this interphase is the phase between two successive M phase, two successive M phase. Okay, and that is what I have written over here. I have written over here, this is the phase between two mitotic phase between two mitotic phase. So I can draw the diagram once again over here. So suppose this is the parent cell, okay, and it is undergoing mitotic phase. M for mitotic phase. M means mitotic phase. Okay, it produces two small daughter cells, and they both of them they undergo interphase. They pass through interphase, and they will produce the mature daughter cells, and they will act as parent cells, and they will produce the daughter cells. They will produce the daughter cells by M phase. Okay, so this is M phase, this is M phase, okay, and this is M phase, okay, and this is interphase. And as I had told you that both these cells they undergo mitotic phase simultaneously. So I will say this interphase is present happening between one M phase and one round of M phase and second round of M phase. Clear? Now M phase is called as most dramatic phase. What is the reason? I have already told you, because the cell organelles most of cell organelles they get reorganized. They get reorganized during this phase. That is why we call this as what? M phase is most dramatic phase. Most dramatic phase. Okay, so here I can write reason. Okay, now so what about the relative durations? Okay, so suppose I have taken a cell, human cell, in the culture medium, and in the culture medium a human cell takes about 24 hours to divide. How long time does it take? 24 hours it takes to divide. So I can say the duration of cell cycle for a human cell in the culture medium is 24 hours. So how long time this interphase will take place? It will take 23 hours. It will take 23 hours in the interphase, and it will take 1 hour in the M phase. That means I can say more than 95% of the duration of what cell cycle is consumed alone by the interphase, and less than 5% of the duration of cell cycle is consumed by M phase. So which one is longer? Interphase is longer. You can see more than 95% duration is consumed by the interphase alone. And what about G1S and G2 phase? G1 requires about 10 hours. Okay, it requires S phase requires about 9 hours, and this phase requires about 4 hours. 4 hours. Okay, so I can say that means I can conclude that what about the decreasing order of the duration of various phases of cell cycle? So I will say G1 phase is the longest phase, then the S phase, then the G2 phase, and then the M phase. In M phase we have different subphases like Prophase, Metaphase, Anaphase, and Telophase. So in M phase, the longest phase is the Prophase. Longest one is the Prophase, then the Metaphase, then the Telophase, and the shortest phase is the Anaphase. Okay, so please remember one thing here. I'm talking about what? I'm talking about the duration, relative duration, not the sequence of phases. If I say sequence of phases, then sequence of phases will be G1 as G2, Prophase, Metaphase, Anaphase, Telophase. But here I'm talking about the decreasing order of duration of various phases of cell cycle. So Anaphase is the shortest phase, G1 is the longest phase. Okay, this is the longest and this is the shortest phase. Okay, now what about the direction? Okay, you can see that you can see the first phase, you have to start with what? G1. So the first phase is G1 phase, then S phase, then G2, then your karyokinesis, then your karyokinesis. So this is karyokinesis. These four phases, they are collectively called as karyokinesis, then cytokinesis, and this karyokinesis and cytokinesis, they are collectively called as M phase. Which phase? M phase. Okay, so I can say these phases, karyokinesis and cytokinesis, they are collectively called as M phase. Okay, so let's solve a question which was asked in the NEET exam. Okay, so during cell growth, DNA synthesis takes place in S phase. So the answer will be S phase. Very simple. Okay, next question you can see. What is the correct sequence of phases of cell cycle? So the first phase will be G1, then S, then M. So this sequence will be G1, S, and G2 and M. Okay, you have to start with the G1, and you have to move in a clockwise manner. Okay, now, and this is almost similar question. Okay, you can see here. Okay, so this is interphase. Interphase will consist of what? Interphase will start with G1 phase. So this will be G1 phase. Okay, this will be S phase. This will be G2 phase. Right? And see how many subphases are there? 4 subphases are there. And these 4 subphases, they will be called as what? Karyokinesis. So this is karyokinesis. And C is cytokinesis. This is cytokinesis. Okay, and what is this? This is karyokinesis. This is nuclear division, right? So this is karyokinesis. Okay, and this is prophase, this is metaphase, this is anaphase, this is telophase. Okay, so I can say that B is basically anaphase. So you can accordingly you can answer this. Okay, and please mention the answer. Okay, now in the comment box. Now, what about the events which take place in the G1 phase, and what about the G1 phase? Okay, so in this diagram you can see clearly that, or let me draw a diagram over here. Okay, so suppose this is cell cycle. This is not a cell, this is a cycle. This is a cell cycle. Okay, and suppose these are the different phases. This is G1 phase, S phase, G2 phase, and this is M phase. Okay, so let me write that: G1 phase, S phase, G2 phase, and M phase. And as I have said that this cycle will take place in a clockwise manner. Okay, so you can see that G1 phase takes place after M phase and before S phase, and that is why I can say that this G1 phase is post mitotic phase. Why? Because it is taking place after M phase, so I can say this is post mitotic phase. This is post mitotic phase and pre-synthetic phase. Because it is taking place before S phase. And what is S phase? Synthesis phase or synthetic phase. So I will say this is pre-synthesis phase. Okay, and I have already told you this is the longest phase of the cell cycle. So I can say this is the longest phase of cell cycle. And the duration of this phase, see, the duration of every phase varies, but the duration of G1 phase varies the most. That is why I will say that this is the most variable phase. That means the duration of this phase will vary most. So let me explain you with example. So if the duration, that means what about the duration of, that means the duration of G1 phase varies. Okay, and the duration of G1 phase, can be short or it can be long. It can be short or it can be long. It can be short as well as it can be long. So when the duration of G1 phase will be short if the cell which is undergoing cell division, it is rapidly dividing. It is rapidly dividing. So in rapidly dividing cell, in rapidly dividing cells, the duration of G1 phase will be short because it will not get enough time to grow. But in case of slowly dividing cells, the cell will have enough time to for the G1 phase. So I will say in case of slowly dividing cells, the duration of the G1 phase will be long. Okay, and as I have told you that in the interphase there will be intense growth, and interphase includes G1, S and G2. So in which phase of the interphase will be the maximum growth? It is the G1 phase, and in this phase most of the cell organelles, their number will be increased. Okay, so I will say in this phase, what are the events which are going to take place? So let me write here events. What are the events which take place in the G1 phase? I will say increase in number of most of organelles. Most of cytoplasmic organelles, their number will increase. Okay, I said most of the organelles. Why? Because the number of Golgi body or mitochondria or chloroplast, they will increase in the G2 phase. Okay, and the centrosome will increase in the S phase. Okay, and in this phase different chemicals are synthesized. So I will say synthesis of amino acids. Okay, amino acids, a number of amino acids will be synthesized. Nucleotides will be synthesized. Okay, and what will be synthesized? It is the nucleic acid, and which nucleic acid? RNA will be synthesized. Okay, and proteins will also be synthesized. So these chemicals are synthesized in which phase? In the G1 phase. Okay, and this is the phase in which maximum growth of the cell will take place. This is the phase in which maximum growth will take place. So I will say maximum growth of cell will take place. Maximum growth of cell takes place in this event. Okay, in this phase, right? So this was about what? This was about the G1 phase. This was about G1 phase. Okay, so what about the S phase then? S phase, why S? Because this is, in this phase DNA synthesis takes place. In this phase DNA synthesis takes place. Okay, and let me tell you one more point. What? Suppose a cell has just crossed the G1 phase. Suppose one cell has just crossed the G1 phase, and once this cell crosses the G1 phase, it will divide at any cost. It will divide at any cost, and that is why whether the cell will divide or not, that decision is taking place in the G1 phase. How? Let me explain you little more. There is a checkpoint at the end of the G1 and the just before the S phase. So this is G1 S checkpoint. This is G1 S checkpoint. Okay, and in this checkpoint, this is G1 S checkpoint. Okay, and if a cell crosses this G1 S checkpoint, it will divide at any cost. It does not matter whether the cell will, whether the cell daughter cells formed are proper or not. It does not matter. But if the G1S checkpoint has been crossed by the cell will divide at any cost. And where do you find this G1S checkpoint? This is present in the end, at the end of the G1 phase. That means if a cell crosses the end of the G1 phase, it will divide at any cost. And if the cell is not supposed to divide, it will not cross this G1S checkpoint. That means it will not cross the it will not enter into the S phase. That means whether the cell will divide or not, the decision is taking place in the G1 phase. So I will write one point. What event is taking? I will say this is the phase of cell division. Phase of cell division is taking place. Is taken. Whether the cell will divide or not, the decision is taking place. Not division. So decision, very sorry. Yes, cell division. Yes. Phase of cell division. Sorry, actually, very sorry. The decision for cell division. The decision for cell division is taken. Okay, now what about S phase? This S phase is known as synthesis phase. This S phase is known as synthesis phase. This is synthesis phase. And in this phase, what happens? What are the events which are going to take place? The events are, in this phase, DNA will replicate. In this phase, DNA replication will take place. DNA replication will take place. And the replication of DNA will increase the amount of DNA but not the number of chromosomes. Why? Because suppose this is one DNA, okay, and suppose this is DNA of chromosome 1, okay, and if it replicates, then what will happen? Two DNA molecules will be synthesized, okay, and they will be held together by a cohesin protein, and these two DNA molecules are now called as one chromosome, not two chromosomes, not two chromatins. So you can see this is one chromatin. Suppose this is chromatin 5. Suppose this is chromatin 5, this is also chromatin 5. In this case, one DNA is present. In this case, two DNA molecules are present. That means DNA number of DNA has increased, but number of chromosome has not increased. Okay, so I will say the DNA replication takes place, and when DNA replication takes place, what happens? Amount of DNA doubles. Amount of DNA doubles, but number of chromosome remains same. It remains unchanged. It remains same. Okay. So let me give you one example then. Suppose there is a cell. Okay. And the cell has, suppose this is a cell, and this cell is present in G1 phase. Okay. And suppose this cell entered into the S phase. Okay, this is S phase. And suppose in G1 phase, the number of chromosome is N. So in the S phase, the number of chromosome will remain N. There will be no change in number of chromosome. Suppose the amount of DNA is C, so the amount of DNA will become double, so it will become 2C. And if the number of chromosome is 2N, okay, so in this case, number of chromosome will remain 2N. But if the amount of DNA is 2C, it will become 4C. So you can see there is increase, there is doubling of the amount of DNA, not the number of chromosome. N is for number of chromosome, C is for amount of DNA. Okay, and if there is a cell which has 3N number of chromosome, so in S phase there will be 3N, but in this case, if the DNA is 3C, it will be 6C. So in this case, you can clearly see that there is increase in the amount of DNA, but not in the number of chromosomes. So what about the other events? Okay. So the other event is during this S phase. What happens? There is synthesis of what? Histone protein. Which proteins are synthesized? Histone proteins are synthesized. And the subunits of kinetochore. You know very well what is kinetochore. We have already learned that in the chapter of cell the unit of life. So the subunits of kinetochore, which is the binding site of what? Spindle fiber, they are synthesized in the S phase. Okay, so this is very, very important. Okay, so histone protein and the unit of kinetochores, they are synthesized in the S phase. And apart from that, in this phase there is duplication of what? Centrosome. The centrosome or the centriole, you can say centrosome or centriole, they duplicate in which phase? It is the S phase. It is the S phase. Okay. So this was about what? S phase. This was about S phase. Okay. Now, what about the, and remember one thing, DNA replication takes place, but if it takes place in the eukaryotic organism, DNA replication takes place in the eukaryotic organism, not the prokaryotic organism, in the S phase, in the eukaryotic cells. So what about the prokaryotic cells? So I will say in prokaryotic cells, PK is prokaryotic. In prokaryotic cells, the DNA replication takes place, it takes place before binary fission. Before binary fission, not in S phase. Okay, not in S phase. This is very, very important. So I can say in E. coli. Okay, because why? Because E. coli is an example of what? Bacteria, prokaryote, right? So if I say in E. coli, in which phase the DNA replication will take place? S phase? No, it is before binary fission. It is before binary fission. Is that clear? Let me check the time and then I will start again. So 1 hour and 1 minute. Okay, no issues. Now, what about the G2 phase? Okay, let me tell you one more point. This G1 phase can also be called as first growth phase or first gap phase. Okay, so I can say this one as first gap phase or first growth phase. These are the different names. Okay, so what about the G2 phase? G2 phase is taking place after S phase, so post synthetic phase, before M phase. So this is pre-mitotic phase. Second gap phase, second growth phase. So let me write those points over here. Okay, let me write those points over here. So what about G2 phase? G2 phase is called as, this is called pre-mitotic phase. This is pre-mitotic phase. Okay, post synthetic phase. Okay, second gap phase and second growth phase. So these are the different names. These are the different names of what? G2 phase. Okay, so what about the events? What about the events which take place during G2 phase? See, in this phase, there is duplication of certain, there is increase in number of certain organelles. So I will say, in this phase, there is increase in number of organelles, certain organelles. Most of the organelles, they show their number, they show increase in their number in G1 phase. Okay, so certain organelles, their number will increase in G2 phase. So what are those organelles? Those organelles are Golgi body. Okay, then which one? Plastid. Plastid means you can take the example of what? Chloroplast and the mitochondrion. Which one? Mitochondrion. Okay, so the mitochondria, plastid and the Golgi body, their number increase in the G2 phase. Okay, and what about centrosome? Centrosome, the number of centrosome increases in the S phase, and other organelles, their number will increase in the G1 phase. Okay, now in the G2 phase, there will be synthesis of proteins also. So it will perform in this phase. There will be synthesis of what? Synthesis of certain proteins like tubulin proteins. Which proteins? Tubulin proteins, which are component of spindle fibers. Okay, which are used in the M phase, and it will also tubulin proteins will be synthesized. Which protein? Tubulin proteins will be synthesized. Tubulin proteins will be synthesized, and also it will synthesize those RNA molecules which are required in the M phase. So I will say RNA molecules, RNAs required in M phase. Those RNA molecules which are required in the M phase, they will be synthesized in the G2 phase. Clear? So this was about G2 phase. This was about G2 phase. Fine. Now, let me tell you one thing. If this is a cell, suppose this is a cell. So these are the different cells. These are the same cell in the different phases of cell cycle. So suppose this is G1 phase, this is S phase, this is G2 phase. Okay. So if this cell is 2n and 2c, that means 2n number of chromosome and 2c amount of DNA are present, then in the S phase the number of chromosome will remain same, but the amount of DNA will become double. And in the G2 phase also the number of chromosome will remain same, but the amount of DNA will become double. Okay. And if this cell is having n number of chromosome and c amount of DNA, then in S phase what will happen? It will be n upon c, so n upon 2c. Okay. And it will be n upon 2c, like this. Okay. So these are the things you have to remember. Okay. Now next move. Let's talk about the G0 phase. Which phase? G0 phase. Okay. So suppose this is a cell cycle. Okay. And suppose this is G1 phase, S phase, G2 phase, M phase. Okay. Let me write those names. G1 phase, S phase, G2 phase, and M phase. Okay. And the cell cycle will start from the G1 phase and it will end with the M phase. It will end with the M phase. So suppose this is the parent cell. And these are the daughter cells. And once the daughter cells are formed, they will immediately enter into the M phase. In the G1 phase. The daughter cells, when they are formed, they will enter into the G1 phase. Okay. So what is this? This is your parent cell. This is parent cells, and what are these? These are the daughter cells. And suppose, as I have told you, that the cell cycle is an energy dependent process, and suppose the daughter cells which are formed just now, they do not have sufficient amount of energy in them. So in that case, the cell will not continue the cell cycle. Rather that cell will exit the cell cycle. This cell will exit the cell cycle and enter into the G0 phase. It will enter into the G0 phase. If energy in the cell is not present in sufficient amount. Okay. And suppose energy is present in sufficient amount, but suppose the cell is not provided with sufficient or proper amount of mitogen. What is mitogen? Any chemical which induces mitosis, which induces cell division. So in the absence of mitogen, the cell will enter into the G0 phase. It will exit the cell cycle and it will enter into the G0 phase. And suppose the cell gets back the so suppose it is again after certain time, suppose the cell which is present in the G0 phase, suppose it gets back, it produces sufficient amount of energy, ATP, and suppose it is provided with mitogen, then in that case it can come back and re-enter into the cell cycle. I did not say always. Why? Because there are certain cells, for example muscle cell or nerve cells. Once they are formed, even though they have enough amount of ATP, even though they are provided with the mitogen, they will not continue the cell cycle. Once they are formed, they will exit the cell cycle and they will enter into the G0 phase forever. They will never come back. That means the daughter cells which are formed, they have two options. They can continue the cell cycle or they can exit the cell cycle. Those cells, once they are formed, which permanently exit the cell cycle and enter into the G0 phase, they are called as terminally differentiated cells. Those cells will once formed, they will never divide, and the example is muscle cell and nerve cells. You can write those points. Okay. But certain cells, they will keep on dividing, and after a certain number of divisions, the telomerase activity will slow down, and because of that they will not divide further, and those cells are called as senescent cell. Which cells? Senescent cells. Third is certain cells, they want to divide, but because of lack of what lack of mitogen or ATP, they do not divide. And if they're provided with mitogen, they will again divide. Those cells are called as quiescent cell. So what are the different reasons for exiting the cell cycle? One is because they are not meant to divide further, like terminal differentiated cell. Second reason is because decrease in the telomerase activity. Third reason is lack of ATP or mitogen. And in the Genome phase, the cell which is present is metabolically active. The cell which is present in the Genome phase, it is metabolically active, it is alive. Okay, but this stage is called as Genome stage, is called as inactive stage. So you should not get confused with this term. Inactive stage it does not mean that the cell will become inactive. No, it is inactive state. But in Genome phase, no activity related to cell cycle will take place, and that is why this stage is called as inactive stage. Okay, metabolic activity will take place, but events related to cell cycle will not take place. That is why this Genome phase is called as inactive stage. And the cell exits the cell cycle from which phase? G1 phase, and enters into the Genome phase. Okay, so let me write those points over here. Okay, daughter cells may exit cell cycle and enter Genome phase. Which phase? Genome phase. Okay, so which phase of cell cycle? From which phase of cell cycle they exit? It is the G1 phase. It is the G1 phase. At the end of G1 phase, they will exit. Okay, they will exit. Actually, at the beginning of G1 phase, but the question was asked in the NEET exam, and in that option, end of G1 phase was given. So the answer will be end of G1 phase. Okay, so this phase is inactive stage. This is called inactive stage. Inactive stage or quiescent stage. This is also called as quiescent stage. So these are the different names of this stage. Okay, but the cell in G0 is the cell which is present in the Genome phase. Genome phase is metabolically active. This is very, very important. This is metabolically active. But this stage is called inactive stage. Why? Because no events related to cell cycle take place. Okay, metabolically active cell. Okay, and the cell which is present in the Genome phase can be of three types. What are those? One is quiescent cell. Okay, let me write here quiescent cell. Second is senescent cell. Okay, SC. And third is terminally differentiated cell. Terminally differentiated cells. Okay, TDC. So you can write those points over here. I have already discussed with you. When the cell enters in, when the quiescent cell enters into the Genome phase, okay, when a cell enters into Genome phase and becomes quiescent cell, if mitogen and ATP they are deficient. What is senescent cell? If the cell is not dividing after a number of divisions, that is called as senescent cell. Why? Because of decrease in the telomerase activity. So I can say over here. And what about terminal differentiated cell? This cell once formed, it will never divide, like muscle cell and the nerve cells. Okay, so if you provide what if you provide ATP and mitogen, ATP and mitogen. What is mitogen? Any chemical which induces cell division, that is mitogen. There will be cell division. Cell division will take place. Cell division will take place. Cell division will take place. Okay. If you provide mitogen and ATP, if you provide ATP and mitogen to what? Senescent cell, there will be no cell division. There will be no cell division. If you provide TDC, terminally differentiated cells, with ATP and mitogen, it will also not show cell division. It will also not show cell division. Okay, let me write over here. This cell has entered into this cell has entered into G0 phase. Why? Because of due to lack of ATP or mitogen or both. ATP or mitogen or both. Why this cell has not is not dividing, senescent cell is not dividing? Due to decrease in telomerase activity. Due to decrease in telomerase activity. Okay. What about the terminally differentiated cells? What about the examples? Muscle cell and which cell? Nerve cells. Nerve cells. And the cell which is present in the G0 phase, it may come back to the cell cycle, re-enter the cell cycle, and it will show further division if it is required. Okay. So I can write over here one point, that the cell present in the G0 phase can re-enter. It can re-enter cell cycle if required. If required, it can re-enter cell cycle. Okay. Now, what about the amount of DNA? And what amount of number of chromosomes? So I will say, I can represent that as 2n upon 2c. 2n upon 2c. 2n number of chromosomes and 2c amount of DNA amount of DNA will be present. Okay. So this was all about what? Interphase. Now we will talk about mitosis. Let me check the time first. Okay, 1 hour and 17 minutes. No problem. See, suppose this is a cell. Okay, and this is parent cell. The cell which undergoes cell division, that is called as parent cell. And after cell division, it will produce two daughter cells. So suppose this is one daughter cell and this is another daughter cell. So these are the daughter cells. Okay. So which cell division is taking place? Mitosis is taking place. Which cell division? Mitosis is taking place. So this is mitosis. Okay. And this cell is parent cell. This is daughter cell and this is daughter cell. These are the daughter cells. Okay. And suppose this cell has n number of chromosomes. So this cell will also have n number of chromosomes and this cell will also have n number of chromosomes. Okay. And if suppose this cell has let me write like this. So if this cell has n number of chromosomes, these daughter cells will also have n number of chromosomes. And if suppose this is the parent cell, okay, and suppose these are the daughter cells. Just a minute. Okay. So suppose this is parent cell and these are the daughter cells. Okay. And what is this? This is parent cell and this is also parent cell. Okay. This is parent cell. Which type of cell division is taking place? Mitosis. Which one? Mitosis is taking place. So after mitosis, we are getting two daughter cells. Suppose in this cell, suppose n number of chromosomes present. Here suppose 2n and here suppose 3n number of chromosomes are present. So in the daughter cells also I will have n number of chromosomes. In this case 2n number of chromosomes, and in this case 3n number of chromosomes will be there. So you can clearly see that the number after mitosis, the daughter cells which are formed, they have the same number of chromosomes as compared to the parent cell, and that is why we call this mitosis as equational division. What do you call this division as? You call this division as equational division. Why do you call this as equational division? Please write that part. Okay, I will dictate you. Equational division. Why? Because the number of chromosome in parent cell is equals to the number of chromosome in daughter cells. Please write that part. And this division is also called as somatic division. Which division? Somatic division. That part also you please write. See, this cell division takes place normally in somatic cells, not in germ cells, normally. And because this cell division is normally taking place in the somatic cell, that is why this cell division is called as somatic division. Please write that part also. Okay, it does not mean that it will never take place in germ cells. It will take place in the undifferentiated germ cells. So I will write one point that undifferentiated germ cells also undergo mitosis. They will also undergo mitosis. So what is mitocyte? Any cell which undergoes mitosis, that is known as mitocyte. So in this case, this is mitocyte, this is mitocyte, and this is mitocyte. So I will say, what is mitocyte? Cell undergoing mitosis. The cell which is undergoing mitosis, that is called as mitocyte. Okay, and what about the nature? This is in case of animal, this is normally somatic cell. This is normally somatic cell. So I will say in animals and in plants. In animals and in plants. Okay, so in animals it is normally somatic cells. It is normally somatic cells. Okay, and this can be haploid, this can be diploid. Okay, let me write here. This is normally diploid. This is normally diploid. This is normally diploid. But haploid cell can also undergo mitosis. Haploid cell can also undergo mitosis. Okay, in plants it can be haploid, it can be diploid, it can be triploid as well. It can be triploid as well. It can be triploid as well. Okay, so this was about what? This was about mitocyte. So I can write one more point over here. In social insects like honeybee, the nature of mitocyte, it is both haploid as well as diploid. It is both haploid as well as diploid. Otherwise, normally in animals, the nature of mitocyte, that means the cell which undergoes mitosis, is normally diploid. But in plant, it is both haploid, diploid as well as triploid also. This was about mitocyte. Okay, what about the mechanism of mitosis? So let's talk about the mechanism of mitosis. And here we are not going to talk about the interphase. We are already done with the interphase. We will talk about the M phase. Before that, let me drink some water. Okay, so let's talk about the M phase of mitosis. Okay, and let me tell you one thing, the cell cycle is a continuous process. The cell division is a continuous process. But for our convenience, we have divided the different events in this cell cycle into different phases. There is no clear-cut distinction between different phases. There is no clear-cut demarcations, or we cannot draw any line between the phases. But for our convenience, we have divided the phases cell cycle into different phases. And what are the phases? In M phase, we have prophase, metaphase, anaphase, and telophase. Okay, this is karyokinesis. And then we will have Okay, cytokinesis. Okay, let me on the switch on the SE first. Okay, so what is prophase? Okay, so the pro word is derived, the pro means first, and phase means stage. Pro means first and phase means stage. So this is the first stage of what? This is the first stage of karyokinesis. Okay, and the NCERT has mentioned the diagram of what? Early prophase, okay, and late prophase, not middle prophase, but we will talk about both early prophase, middle prophase and late prophase. Okay, so the prophase can be divided into three subphases: early prophase, okay, then middle prophase and late prophase. So what are going to happen in this early prophase, middle prophase? We will talk about that in detail. So suppose this is an animal cell. Okay, and this animal cell will have plasma membrane and nucleus. So this is the nuclear envelope. Both membranes are present. Two membranes are present. So this is the nuclear envelope and this is the plasma membrane. Okay, now in this prophase, what will happen? We know the sequence of phases G1, then S, and then G2. Okay, then the prophase will take place. And the prophase will also be, it will be early prophase. See, in these phases, the DNA is present in which form? In the form of chromatin. And in the prophase, the DNA, the chromatin will start to undergo condensation. It will become condensed to form the shorter and thicker chromosomes. Okay, chromatin is longer structure and thin structure, whereas chromosome is thick and short structure. Okay, so I will say, so what is the event which is going to take place in the early prophase? I will say chromatin will undergo condensation to form chromosome, but this condensation will not get completed. The condensation will start in the early prophase and end by the metaphase. So I will say starting of condensation starts. Okay, and see, more is the condensation, the chromosome will be thicker and shorter. Okay, and because the condensation has just started, that is why the chromosome which has just formed is thinnest chromosome. This is the thinnest chromosome and this is the longest chromosome. Thinnest chromosome and longest chromosome. Okay, so suppose this is a chromatin. If I condense it, if I start to condense it, it will become thicker and shorter. Okay, if I do further condensation, it will become more thicker and shorter. If I condense it further, then it will become more thicker and shorter. Okay, so what is happening? With condensation, the thickness is increasing and the length is decreasing. And because the condensation has just started, that is why the chromosome will be what? The chromosome will be thinnest and longest. And these chromosomes, see, in the S phase, the DNA has replicated, and both the DNA molecules in the same chromatin will further form the chromatids. We have studied this already studied in the last lecture, that is Cell the Unit of Life. You can watch that part. Okay, so in this case, condensation has started, but the chromatids are not visible. This is important, so I will say these chromatids are not visible. Chromatids are not visible. They are not visible yet. Okay, and these chromosomes, they will be their end will be bound to the what? Nuclear membrane. Okay, and their end, that means telomere will not be visible. The telomeres will not visible. Okay, so I will say one more point, that telomeres, which one? Telomeres are attached. Okay, you can leave this point, no problem. Telomeres are not visible. Okay, and the chromosomes, they appear as a ball of wool. If I give you a ball of wool, it is difficult for you it will require time to find out the end of the wool, right? So in this case also, if this is nucleus, okay, and suppose this is these are the chromosomes. Suppose this is one chromosome, this is another chromosome, okay, this is another chromosome. So suppose these are the chromosomes. Suppose these are the chromosomes. Okay, so this appears as a ball of wool. Why it appears as a ball of wool? Because we cannot say this is the end of this chromosome, this is the end of this chromosome. We cannot say that. Okay, and that is why we can call this structure as the entire chromosome complex as what? We can compare that with a ball of wool, and that is why this stage is called as what? This stage is called as what? This stage is called as spireme stage. Which stage? Spireme stage. So I will say, and that is why early prophase is called spireme stage. Which stage? Spireme stage. Okay, and in this phase, what else are going to happen? See, on the outer side, okay, on the outer side of the nucleus there will be centrosomes, and in the S phase what has happened? The centrosome has duplicated, so there will be two centrosomes. So suppose this is one centrosome, this is another centrosome. Okay, and now what will happen? Both these centrosomes, they will move away from each other. This will move in this direction and this will move in this direction, and when they start moving, they will produce certain ray-like structures, and those ray-like structures are made up of microtubules, and those structures are called as astral rays. So here what will be formed? Astral rays will be formed. So these are the astral rays. These are astral rays. Okay, and now centrosome plus astral rays, they are collectively called as ester. And you can clearly see that in animal cell, how many esters are present? Two esters are present. This is one ester, this is another ester, and that is why I call this as amphiestral cell division. Okay, cell division in which ester is formed, that is astral cell division. If ester is not formed, anastral cell division. In plant cells, we do not have centrosome. If there is no centrosome, there will be no ester. Okay, why? Because centrosome plus astral ray, collectively called as astral ray, ester. In plants, there is no centrosome, so there will be no ester. So plant cell division is called as anastral cell division, anastral. But animal astral cell division, how many esters? Two. So I will call that as amphiestral cell division. Which one? Amphiestral cell division. Okay, so I will label it like this is basically astral ray. Astral ray. Okay, and what is this? These are the centrosomes. These are the daughter centrosomes. Okay, and what else? In this phase, what will happen? The nucleolus in the nucleus will start to disappear. Okay, so let me write that part also. Okay, so suppose this is the nucleolus. Now it will start to disappear. So I can write here that this is disappearing nucleolus. The nucleolus will start to disappear. Okay, and in this phase, what else will happen? The endoplasmic reticulum and Golgi body, they will also start to disappear. Which one? Endoplasmic reticulum and Golgi body, they will start to disappear. Why? We know that endoplasmic reticulum is a scattered structure, and in anaphase we have to separate the chromosomes in two different poles, and to separate the chromosomes in two different poles, the space should be free. Okay, that means some chromosomes will move in this end and some chromosomes will move to this end. Okay, and we know the endoplasmic reticulum will be present here, it will be present here. So if there is endoplasmic reticulum, then the separation of chromosome will be interfered, and not to interfere the separation of chromosome, the endoplasmic reticulum and the Golgi body, they have to be disappeared. they have to be disappeared. Okay, so and their disappearance will start in the early prophase. And remember one thing, nucleolus and endoplasmic reticulum Golgi body, they start to disappear in the early prophase, but they will completely get disappeared in the late prophase. Okay, so here I will have endoplasmic reticulum and I will also have Golgi body. Number of endoplasmic reticulum and number of Golgi body will be there. Okay, now those points I have written over here. So okay, movement of daughter centrosomes will take place. Okay, and astral rays will be formed, and these astral rays are made up of what? They are made up of microtubule. They are made of microtubules. Okay, and what is aster? Aster is basically one. Aster is made up of what? Astral rays plus centrosome. Astral rays plus centrosome. Okay, and what about cell division in animal? In animal, the cell division is astral. Why? Because aster is formed. And what type? To be specific, amphiastral. Why? Because two esters are formed. What about cell division in plant cells? Because there is no ester formation, so I will say anastral cell division. Anastral cell division. Okay, and what else events are going to take place? I will say nucleolus will start to disappear. Nucleolus will start to disappear, and endoplasmic reticulum, Golgi body, they will also start to disappear. This was about what? Early prophase. What about middle prophase? In the middle prophase, there will be further condensation of chromosome. Okay, and I can tell you one more point, that for KBPI students I am talking about, this condensation of chromosome is due to the condensin protein. This is for KBPY students. Okay, condensin proteins. So condensin proteins, they perform condensation. Okay, now in the middle prophase, the condensation continues. Okay, and now the chromosomes have become little thicker and little shorter, and in this case, in this phase, now the chromatids will become visible. So I will say chromatids are visible. Okay, and if you look at the chromosomes, you can clearly see that two chromatids of a same chromosome, which are called as sister chromatids, they are held together at the centromere. By the help of cohesin protein. Okay, so I can say that two sister chromatids, they appear to be held in a single chromosome. They are held in a single chromosome at centromere. By whom? By cohesin protein. So cohesin protein will be holding these two sister chromatids. Okay, so let me draw that diagram over here. So suppose this is the nuclear envelope. Okay, and suppose this is one chromosome. Okay, and this is another chromosome. These are the chromosomes. Okay, so these lines, this is one chromatid, this is another chromatid. Okay, and those chromatids are held, they are held by cohesin protein. Okay, so if this is chromatid, this is another chromatid, there will be cohesin protein in between them. So cohesin protein will be there. I am not mentioning that over here. Cohesin protein will be there. Okay, and the portion at which they are held together, that area is called as centromere. So I will say, suppose this is the centromere. And this is centromere. These are the centromeres. Okay, and suppose this is chromosome 1, this is chromosome 1, this is chromosome 2, and this is chromosome 2. Okay, the size of chromosome 1 is larger as compared to the size of chromosome 2. And in this case, what will happen? In this case, the nuclear membrane Which one? Nuclear membrane, it will start to disappear. It will start to get disappear. It will not disappear completely. Okay, and nucleolus will also be there. Okay, nucleolus is also there, and it has started to disappear. Okay, and it will keep on disappearing. It will keep on disappearing. So in this case, the size of nucleus will be shorter or smaller as compared to the early prophase. Which prophase? Early prophase. So this is the nucleolus. This is the nucleolus. Okay, so what are the events which are going to happen in the middle prophase? So I will say what? Nuclear envelope starts to disappear. It starts to disappear. Okay, and in this middle prophase, these daughter centrosomes will have reached to some distance. Okay, so let me write that part. Okay, so here let me draw that part. So suppose this is one centrosome. Suppose this is one centrosome, this is another centrosome. Okay, and they are producing what? They are producing the, suppose they are producing the estrol rays. Suppose they are producing the estrol ray. So suppose this is estrol ray. Okay, and this is the estrol ray, and they will keep on moving. They will keep on moving. Okay, and Golgi body will be there. Endoplasmic reticulum will also be there. Okay, nucleolus starts to dissipate in the early prophase. Golgi body and endoplasmic reticulum, they also start to dissipate in the early prophase, but nuclear membrane starts to dissipate in the middle prophase. What about late prophase? In the late prophase, what will happen? Further condensation will take place of the chromosome. So the chromosomes will become further shorter and thicker. Okay, so suppose, let me draw those chromosomes over here. So suppose this is one chromosome. Okay, this is another chromosome 1. So these are the chromosome 1, and there is centromere. So this is centromere. Okay, and let me draw the chromosome 2. So suppose this is chromosome 2, and suppose this is chromosome 2. And here also there will be centromere. So suppose this is centromere, suppose this is centromere. Okay, chromosome 1, chromosome 2, chromosome 2. And in the late prophase, what will happen? Condensation will continue. What will happen here? Condensation will continue. Condensation of chromosome continues. Okay, and the second thing which happens is, in this phase, nuclear envelope will completely get disappeared. And when nuclear membrane will completely disappear, at the end of late prophase, what will happen? These chromosomes will come and lie into the cytoplasm. So I will say over here, what happens? There is complete disappearance of what? Endoplasmic reticulum, Golgi body, and nucleolus and nuclear membrane, or I can say nuclear envelope. Okay, and when the nuclear envelope disappears completely, what will happen? The chromosomes, they come and lie in the cytoplasm. Okay, and the centrosomes present over here, they have reached the opposite poles over here. Both of them, they have reached the opposite poles, and they have formed the estrol rays. So these are the two esters. So two esters have reached the opposite poles. And now what will happen? Spindle fiber formation will take place. What will take place? Spindle fiber formation will take place. So this is spindle fiber. These are the spindle fiber. Their formation have started. These are the spindle fibers. Okay, so I can write that this is spindle fiber. For spindle fiber, I am writing SF. Okay, so I can say what happens? Spindle fiber formation takes place. Spindle fiber formation takes place. Formation starts. Okay, there is a term. So what is this? This is late prophase. Okay, there is a term which is called as mitotic spindle. mitotic spindle. So I will say one point, that formation of mitotic spindle or mitotic apparatus. Formation of mitotic apparatus will take place. Okay, takes place. So what is mitotic apparatus? Mitotic apparatus consists of various components like what? Like ester plus, ester means what? Centrosome plus spindle fiber, astral rays plus spindle fiber. Ester plus spindle fiber collectively called as mitotic apparatus. They are collectively called as mitotic apparatus. Okay, so this was about early prophase, so late prophase. So prophase has completed. Now what about, before discussing about what transition to metaphase, we should learn about transition to metaphase. Transition to metaphase. Now what will happen? So suppose this is chromosome. So suppose this is one chromosome. Okay, suppose this is another chromosome, chromosome 1, and suppose this is one chromosome, and suppose this is another chromosome. These are the different chromosomes. Okay, so this is chromosome 1, chromosome 1, chromosome 2, and chromosome 2. Okay, and now suppose this is one centrosome, this is another centrosome. Okay, and suppose these are the astral rays, these are the astral rays. Okay, and now what will happen? The spindle fiber, they will go and bind to the chromosome. They will go and bind to the chromosome. Okay, so this will go and bind to the chromosome. Which one? Spindle fiber will go and bind to the chromosome. And you can clearly see that each chromosome is attached to the spindle fiber coming from two different ends. And remember one thing, if this is a chromosome, okay, so this part will be centromere, this part will be centromere. Okay, and on the either side of the centromere there will be some disc-like structure. There will be disc-like structure, and what do you call this disc-like structure? Kinetochore. Okay, and to this kinetochore the spindle fiber will get bound. So this is spindle fiber. These are the spindle fibers. Okay, and what is this? This is kinetochore. This is kinetochore, and what is this? This is centromere. This is centromere, and what are these? These are the sister chromatids. These are the sister chromatids. Okay, so this is what happens, and some spindle fibers will reach to the opposite poles. They develop at the one point and they will reach at the opposite poles, and some will start, but they will not reach to the opposite pole. They will neither get bound to the chromosome, not with the what, nor they will reach the opposite poles. Okay, and the spindle fiber which is bound to the chromosome, that is called as chromosomal spindle fiber. Spindle fiber which is bound to the chromosome is known as chromosomal spindle fiber. Spindle fiber which originates one pole and reaches the opposite pole, that is called as continuous spindle fiber. And this spindle fiber which is not bound to any chromosome, okay, and has not reached to the opposite pole, that is known as discontinuous spindle fiber. So how many spindle fibers? Three types: chromosomal, continuous, and discontinuous. Three different types of spindle fibers are present. Okay, so let me label them. Okay, so this is continuous spindle fiber, okay, this is discontinuous spindle fiber, okay, and this one is chromosomal spindle fiber. Chromosomal spindle fiber. And remember one thing, each chromosome is bound to the spindle fiber coming from two different ends. Okay, now what about metaphase then? In metaphase, what will happen? The spindle fibers present over here, they will undergo contraction and expansion. That means they will undergo what? Depolymerization, shortening, and polymerization, that means elongation. And because of that, all these chromosomes which are scattered in the cytoplasm, they will come in a single plane. In a single plane they will come. Okay, and that process of bringing all the chromosomes in a single plane is called as what? Congregation. What do you call that? Congregations. Okay, so in metaphase what happens? Congregation will take place. And another event which is going to happen is in metaphases is there will be complete condensation of chromosome. Complete condensation of chromosome. So I will say 2 points NCERT says, you can write that point. The beginning of metaphase is marked by the complete disappearance of what? Nuclear envelope. You can write that point. Second point is that condensation of chromosome completes by metaphase. Okay, so let me write those points here. Okay, complete disappearance of what? Complete disappearance of nuclear envelope. It marks the beginning. It marks the beginning of what? Beginning of metaphase. Beginning of metaphase. Second thing is that condensation of chromosome. Condensation of chromosome completes. Okay, and that is why the chromosomes in the metaphase, they are shortest and thickest. In which phase? Metaphase. In metaphase the chromosomes are shortest and thickest, and because of that their morphology can be easily observed and their number can be easily counted. Okay, so I can say that, and that is why I can say that this is the phase, this is the best phase, this is the best phase to count number of chromosomes. And study morphology of what? Morphology of chromosomes. So the morphology of chromosomes can also be studied in this phase. This is the best phase to study. And if I say morphology, that means it is talking about what? Size. Okay, it is talking about shape also. It is talking about shape. Okay, and actually we should prefer for shape, we should prefer Anaphase. Okay, but if number, size, shape all are given together, then you should prefer the metaphase. You should prefer the metaphase. Okay, so this was about something about metaphase. And what happens in this phase? Congregation takes place. What happens? The event which happens in this phase is congregation. Which one? Congregation takes place. So what do you understand by congregation? Congregation means bringing of all chromosomes together. Bringing of all chromosomes at spindle equator. At spindle equator. If you are bringing all the chromosome at spindle equator, that process is called as congregation. So because of congregation, what is formed? Metaphasic plate is formed. So I will say it will cause the formation of metaphasic plate. Formation of metaphasic plate. Okay, and this metaphasic plate is also called as equatorial plate. What do you call this one? As equatorial plate. Okay, so what is this equatorial plate or metaphasic plate? This is basically nothing but the what? Plane of alignment of chromosomes. In which phase? In metaphase. In metaphase, the plane in which the chromosomes are aligned is called as metaphasic plate. And how many metaphasic plate are formed? Only one metaphasic plate is formed. How many? One in metaphase. In metaphase first, two, metaphase second, one metaphasic plate is formed. Okay, so let me draw the diagram so that you can understand that easily. So suppose this is the cell, animal cell. Okay, and suppose these are the chromosomes. Suppose these are the chromosomes. Okay, and let me draw the centromere over here. So suppose this is the centromere, this is centromere. So these are the centromeres, okay, and you can clearly see that the centromeres are lying in the same plane. Okay, but the arms are oriented differently. This is one arm, this is another arm. So these arms are oriented in different ways, but the centromeres are present in a single plane, and this plane at which the centromeres lie, that is called as metaphasic plate. And let me draw the spindle fiber over here. So suppose this is the centrosome. Okay, and suppose these are the spindle astral rays. These are the astral rays. Okay, and let me draw the spindle fiber. So this is spindle fiber, these are the spindle fibers. These are the spindle fibers, different types of spindle fibers. Okay, these are the spindle fibers. Okay, so this is what happens. So you can clearly see one thing, that this is chromosome 2, this is chromosome 2, this is chromosome 1, this is chromosome 1. So larger chromosome lies at the center and smaller chromosomes lies at the periphery. Okay, so in metaphasic plate, which point you have to remember? For KBPI students, I am writing this point. Okay, so in metaphasic plate, smaller chromosomes, they lie at periphery, and larger chromosomes, they lie towards the center. Okay, so this was about metaphase. So in metaphase, the main points you have to remember is congregation. That means bringing of all chromosomes at the center. Okay, you can see in transition to metaphase, the chromosomes were scattered, but in metaphase, what happens? All chromosomes, they are brought at the same plane. Okay, and that forms the metaphasic plate. What do you call the metaphasic plate? We call this metaphasic plate as equatorial plate. How many? One metaphasic plate is formed, and in metaphasic plate, smaller chromosomes lie towards the periphery, and larger one, they will lie at the center. Okay, and what is metaphasic plate? Plane of alignment of chromosomes. Now what happens? Now in this case, in anaphase, what is going to happen? The sister chromatids, they will get separated. Okay, so let me draw one diagram over here. Okay, so suppose this is the this is a cell. Okay, and now what is going to happen is, suppose this is one sister chromatid. Suppose this is one sister chromatid, this is another sister chromatid. Okay, and now suppose this is one spindle fiber, this is another spindle fiber. Okay, and in between them, what is present? Cohesin protein is present. Now this cohesin protein will break, and this cohesin protein is responsible for holding these two sister chromatids. So if this cohesin protein breaks, then what will happen? This sister chromatids will become free, and this spindle fiber will pull this chromatid, and this spindle fiber will pull this chromatid towards this end. Okay, and that is why they will move away from each other, and after some time you will see a structure like this. So these are these two sister chromatids. Okay, and this is the spindle fiber, and this is the spindle fiber. Okay, so I can say this chromosomal spindle fiber is pulling this chromatid. This chromosomal spindle fiber is pulling this chromatid. And once they are pulled away from each other, then what will happen? Another type of spindle fiber will be formed in between them. And these spindle fibers will keep on increasing. So because of elongation of these spindle fibers, these spindle fibers will now push this two chromatids. That means I can say there is pulling of chromosomes as well as sister chromatids and pushing of sister chromatids away from each other. Okay, pulling by this chromosomal spindle fiber and pushing by this interzonal spindle fiber. These spindle fibers are called as interzonal spindle fiber. Okay, so what is happening in this phase? In anaphase, the sister chromatids, they get separated. So I will write over here what happens in this phase. In anaphase, what happens? Centromere splits into two. Okay, so one centromere become two centromere, and one centromere will come here and another centromere will come here. So this one is called as now daughter chromosome and this one is called as now daughter chromosome. They are called as daughter chromosomes. Okay, so these are the daughter chromosomes. And what else happens in this phase? In this phase, the cohesion protein is degraded or it is broken. Okay, and when the cohesion protein is broken, what will happen? The sister chromatids will become free, and as a result, because of these two things, what will happen? The two sister chromatids become free. They become free, and they are now called daughter chromosomes. They will be called as daughter chromosomes now. Okay, and what else happens in this phase? There is separation of daughter chromosomes. Okay, and previously they were sister chromatids, so I will say separation of sister chromatids will take place. And this separation takes place due to two different things. One is by pulling and second is by pushing. Okay, pulling by whom? Chromosomal spindle fiber. Okay, and pushing by interzonal spindle fiber. Okay, so let me draw that diagram over here. Okay, so suppose this is centrosome. This is centrosome. Okay, and the shape of the chromosome is actually observed in this phase. Actually, it is observed in this phase. Okay, and suppose these are the astral rays. Okay, these are the astral rays. Okay, now let me draw the chromosome over here. So this is one chromosome, daughter chromosome. This is another daughter chromosome. This is one daughter chromosome. These are the daughter chromosomes. These are the daughter chromosomes. Okay, and this is centromere. These are the centromeres. Okay, and here kinetochore will also be there. Which one will be there? Kinetochore will also be there. So here let me draw the kinetochore. So suppose this is kinetochore. This is kinetochore. This is kinetochore. These are the kinetochores. These are the kinetochores. Okay, and to this kinetochore, what will be bound? The spindle fibers will be bound. Okay, so suppose these are the spindle fibers. These are the spindle fibers. Okay, these are the spindle fibers. These are the different types of spindle fibers. Now, in between these, some other spindle fibers will also develop, and these spindle fibers are called as interzonal spindle fibers. These are called as interzonal spindle fibers. Between two chromosomes, daughter chromosomes will be there. These are the interzonal spindle fibers. Okay, and this is daughter chromosome 2, daughter chromosome 2, daughter chromosome 2, daughter chromosome 2, daughter chromosome 1, 1, daughter chromosome 1, daughter chromosome 1. So in this case, I can say that I can say this condition can be called as what? How many copies of DNA 1 is present? 4 times 1, 1, 1, 1. So 4 times. So I will say 4C. 2 is also present 4 times. So I will say 4C. What about number of chromosome? Number of each chromosome is present 4 times. 1 is present 1 time 4 times. 2 is present 4 times. So I will say 4n upon 4C. So in case of Anaphase, the number of chromosome will be 4n and the amount of DNA will be 4n. Sorry, 4C. Okay, so this is what I will write. Okay, so this was about what? This was about the Anaphase. This was about Anaphase. Okay, so I can write one point over here. Prophase, Metaphase, and Anaphase. In prophase, number of chromosome is 2n and amount of DNA is 4C. Number of chromosome is 2n, amount of chromosome is 4C. But in Anaphase, number of chromosome is 4n and amount of chromosome is 4C. So in which phase the number of chromosome becomes double? It is the Anaphase. Okay, see, from 2n it is 4n. So I will say in Anaphase, number of chromosome becomes double. So I will write one point over here. Anaphase, number of chromosome doubles. Number of chromosome doubles. Okay, so and what is this? This is interzonal spindle fiber. This is interzonal spindle fiber. Okay, and let me tell you one more point over here. That in Anaphase, just a minute. Let me take 1 minute. Let me check the time. 2 hours. 2 hours, 6 minutes. No issues. One question was asked in the previous years. Okay, so that is why I'm writing here. So there is a complex which is called as Anaphase promoting complex. Which one? Anaphase promoting complex. Okay, so Anaphase promoting complex is also known as APC. What do you call that as APC? So let me write here. There is a complex which is called as MPF. Which one? MPF. Okay, so what is MPF? This is MPF is maturation promoting factor. What do you call this as? Maturation promoting factor. So this maturation promoting factor, it activates Anaphase promoting complex. And this Anaphase promoting complex will inactivate the Anaphase inhibitor. It will inactivate the Anaphase inhibitor. Okay, and when the Anaphase inhibitor is inactivated, what will happen? Anaphase will start. Anaphase starts. Okay, that means I will say in the prophase and metaphase, Anaphase inhibitor is active. In prophase, during prophase and metaphase, Anaphase inhibitor is active. Okay, and that is why Anaphase is not taking place. The separation of sister chromatid is not taking place. But once the Anaphase comes, what happens? MPF will activate Anaphase promoting complex. Anaphase promoting complex will inactivate. It will inactivate the Anaphase inhibitor. And once the Anaphase inhibitor is inactivated, what will happen? The Anaphase will start. Okay, and what about the example of Anaphase inhibitor? Okay, APC is Anaphase promoting complex. This is Anaphase promoting complex. Okay, this is Anaphase promoting complex. Fine. So let me write one example over here. Okay, Anaphase, one of the examples of Anaphase inhibitor is securin. Which one? Securin. Okay, so suppose this is securin. Okay, and suppose this is separase. Suppose this is separase. Okay, so this is separase. This is separase. This is separase. Okay, and this is securin. And this securin is in active state. And when this is in active state, it will bind with the separase. And when this separase is bound to the securin, it is in inactive state. It is in inactive state. When the securin, which is an example of what, anaphase inhibitor, it is in active state, it will bind with the separase, and when the separase is bound with the securin, it is in inactive state. Okay, and suppose APC has come. Which has come? APC has come. So this APC will inactivate the securin. It will inactivate the securin, and that is why the securin will become inactivate. And once the securin is inactive, it will not bound with the separase. So what will happen? The separase will get separated. So the securin will get separated. So I will say this is securin. This is securin, and in which state it is present? In inactive state is present. Inactive state is present. Okay. Now this separase. Now this separase is active. Now this separase, because it is not bound, with the securin, that is why now this is active. So what is this separase is going to do then now? Let me tell you. When this is active, what is going to happen? So suppose this is one chromatid. Okay, this is another chromatid. They are held together by whom? By the help of cohesin protein. So suppose this is cohesin protein. This is cohesin protein. Now what will happen? This separase will break down the cohesin protein. This will break down the cohesin protein. So this is cohesin protein. Which one? Cohesin protein. So in this case, this separase will break down the cohesin protein, and that is why these two sister chromatids now will become free. Okay, so and because they are free, so I can write here like this. So this is one sister chromatid, this is another sister chromatid. Now they are free from each other. Now they are free from each other, and now they are called as daughter chromosomes. Okay, so you can write here breakdown of what? Breakdown of cohesin protein. Okay, now this was about anaphase. So what is happening? And you can also write one point here. You please write one point over here. Okay, so if this is chromosome, so this is the telomere, this is the telomere, and this is the centromere. So what is happening? So while during movement, the telomeres are facing towards the center, and the centromere is facing towards the pole. Telomere is facing towards the pole. Okay, so you can write that point also here. Okay, so in this phase, what is happening during this movement or during this separation? The telomeres or the arms, they face towards the center. They face towards the center, whereas the centromere of chromosomes during this movement, they face towards the periphery. Periphery means towards the pole. Okay, now what about telophase? In the telophase, what will happen? The chromosomal group will reach the opposite poles. Okay, and once they reach the opposite poles, so suppose this is SL. Suppose this is SL. This is SL. Okay, and suppose in this case, chromosome 1 has reached chromosome 2 copies of chromosome 1 and 2 copies of chromosome 2 have reached. Here also 2 copies of chromosome 1, 2 copies of chromosome 2 have reached. Okay, now what will happen? Around this chromosomal group, one nuclear envelope will develop. Here also nuclear envelope will develop. Here nucleolus will reappear. Here nucleolus will reappear, and this chromosome will undergo decondensation to form the chromatin. And here what do I have? I have the what spindle fibers are also there. Okay, here spindle fibers are also there, and these spindle fibers will disappear, and astral rays will also disappear. That means all the events which take place in the prophase, just opposite events will take place. So I will say this phase is basically what? This phase is opposite to early prophase. This is opposite to prophase. Okay, so let me write here. So here what will happen? Nuclear envelope will reappear. Here also nuclear envelope will reappear. Okay, and what is this? Suppose this is centrosome. This is centrosome. Suppose this is nuclear envelope. This is nuclear envelope. Okay, and in this case what will happen? The nucleolus will reappear. Nucleolus will reappear, and the chromosomes which are present, they will undergo decondensation to form the chromatin. Okay, so suppose these are the chromatins. These are the chromatins. Okay, these are the chromatins. These are the chromatins. Okay, and here what do I have? I have, suppose this is the centrosome. This is the centrosome. Okay, and here astral rays are there. Here astral rays are there, and these astral rays will disappear. Spindle fibers will also get disappeared. Spindle fibers will also disappear. Okay, and apart from that, endoplasmic reticulum and Golgi body, endoplasmic reticulum and Golgi body, they will reappear. They will reappear. Okay, so I will say this is basically reverse of prophase. Reverse of prophase. So what are the events which are going to take place? So the events which take place are what? Reappearance of what? Nucleolus, endoplasmic reticulum, Golgi body, and nuclear envelope, or I can say nuclear membrane. NM is nuclear membrane. Okay, so all these things, they reappear, and then spindle fiber and the astral rays, they disappear. And in case of plants, you can write that point. In case of plant, interzonal spindle fiber will persist. Interzonal spindle fiber will persist, and the chromosomes, they undergo decondensation to form the chromatin. And looking at the chromatin, you cannot say this is the chromatin 1, chromatin 2. That means the chromatin will, that means the chromosome will lose their identity. The chromosome will lose identity. So I will say the chromosomes, they lose their identity. Looking at the chromosomes, you can say, okay, this is chromosome 1, chromosome 2, or chromosome X, or chromosome Y. But by looking at the chromatin, you cannot say this is chromatin 1 or chromatin 2 or chromatin X or chromatin Y. You cannot say that. Okay, so what is this? What is happening over here? This is the decondensation. Decondensation takes place. Okay, so this is telophase. Okay, now what happens after telophase? Cytokinesis will take place. What is cytokinesis? Cytokinesis is basically division of cytoplasm. In normal case, the cytokinesis takes place after the karyokinesis, but in some cases, in the middle of the anaphase, it will start and it will end at the telophase. It will end with this telophase. Okay, so in cytokinesis, what happens? There is division of cytoplasm. So let me write that point. Normally starts after karyokinesis. After karyokinesis, it will start, but in some cases it will start at the mid of anaphase and it will end with the end by the telophase. So, but I will say, but in some cases, but can start at mid anaphase and end by the end of and at the end of telophase. That means it ends with karyokinesis. Okay, and cytokinesis cytokinesis is of two types. It is of two types. So I will say this is of two types. What are those? One is furrow formation method. One is furrow formation method. Okay, and second is your cell plate formation method. Cell plate formation method. Okay, and this furrow formation method takes place in which organism? Animal cells. In animal cells, the cytokinesis is of what? Furrow formation method. And in plant cells, and if I say plant cells, that means higher plant cells. Which plant cells? Higher plant cells. So that means in lower plant cells, furrow formation will take place. And whenever the question is asked plant, you have to think about higher plant cells. Which plant cells? Higher plant cells. Clear? Now in some cases what happens? Suppose this is a cell, and suppose the karyokinesis has already taken place. So suppose this was the cell and this was the nucleus. After karyokinesis there will be two nuclei. Okay, and suppose in this condition there is no cytokinesis, so this cell will be binucleated cell. And suppose this cell again undergoes karyokinesis, so this will produce 4 nuclei. And suppose there is no cytokinesis, so what is happening? Multinucleated condition will develop. Okay, and the cell division in which only karyokinesis takes place but not cytokinesis takes place, that type of division is known as free nuclear division. What do you call that? As free nuclear division. So let me write that part over here. Okay, so free nuclear division. So what do you understand by free nuclear division? In free nuclear division, karyokinesis takes place but cytokinesis does not take place. Karyokinesis takes place, so K plus means karyokinesis is taking place, C minus means cytokinesis is not taking place. Okay, that is known as free nuclear division. Okay, and what is the result? It results in multinucleate condition. Multinucleate condition. And this multinucleate condition is called as syncytium. And the example is in coconut milk. Coconut milk consists of a number of nuclei, free nuclei. Okay, it consists of a number of free nuclei. Okay, and this coconut milk, it actually represents free nuclear endosperm. It represents free nuclear endosperm. Okay, in class 12 we will learn about that. So let us talk about these methods one by one. So suppose this furrow formation method takes place in animal cells. So suppose this is the plasma membrane. This is the plasma membrane. Okay, this is the plasma membrane. And suppose these are the daughter nuclei. So suppose this is one daughter nucleus, this is another daughter nucleus. So these are the daughter nuclei. For nuclei I am writing N. Okay, now what will happen in the next step? In the furrow formation method, the first thing which is going to happen is there will be formation of what? There will be formation of, so let me draw that first. These are the nuclei. So the first thing which is going to happen is, in this case, a contractile ring will develop. A contractile ring will develop and it will be bound with the what? Plasma membrane at two ends. It will be bound with the plasma membrane at two ends. So this is the contractile ring. So this is a ring-like structure and this is bound to what? Plasma membrane. This is contractile ring, and this contractile ring, it has microfilament. It has microfilament. MF is microfilament. What is this MF? MF is microfilament. It contains microfilament. Now what will happen? This contractile ring will undergo contraction. It will undergo contraction. Okay, and because this contractile ring is attached to the plasma membrane, this is attached to the plasma membrane, and because the plasma membrane is elastic, that is why this plasma membrane will also undergo contraction. When this contractile ring contracts, the plasma membrane will also undergo contraction. Okay, and that is why a structure like this will be formed. So suppose this is the contractile ring. Suppose this is the contractile ring. Okay, and suppose this is the plasma membrane. This is the plasma membrane. Okay, and suppose these are the nucleus. So this is nucleus, this is nucleus. Okay, these are the nucleus. Okay, and in this case, you can see there is a depression. There is a depression, and these depressions are called as furrow. So this is furrow. So this is furrow. So due to the contraction of contractile ring, what is formed? Furrows are formed. Two furrows are formed. And now what happens? Now these contractile rings, they will further undergo what? Further undergo contraction, and because of that, this furrow will develop in this direction, and this furrow will develop in this direction. That means it will deepen, and this will also deepen in this. That means there is centripetal development of furrow. There is centripetal development of furrow. Okay, that means I can say after certain time, the diagram will look like this. This is contractile ring, right? And this is the plasma membrane. This is the plasma membrane. Okay, and these are the nucleus. These are the nucleus. So this is centripetal development of furrow. Furrow is developing in growing centripetally. So I will say centripetal growth of furrow. The furrow shows centripetal growth. Okay, and like this, finally what will happen? These two furrow will fuse with each other. The two furrows will fuse with each other. So this membrane will fuse with this membrane, and this membrane will fuse with this membrane. So ultimately I will get two cells. So this is one cell, this is another cell. So these are the two cells. Okay, and they will have the nucleus. And these cells will be called as daughter cells. So this is furrow formation method. This is contractile ring. So the first event which is going to take place is appearance of contractile ring. Second, contraction of contractile ring, which will lead into the formation of what? Furrow. And then deepening of the furrow. That means centripetal growth of the furrow. Further centripetal growth of the furrow. So like this will take place, and ultimately it will produce the two daughter cells. This was about furrow formation method. And what about cell plate formation method? This takes place in the plant cells. Why furrow formation method does not take place? Because in plants we have cell wall, and cell wall is rigid, and this cannot enter, this cannot allow the formation of furrow. And that is why there should be another method in plant cells, and the method is cell plate formation method. So cell plate formation method, it takes place in which organism? It takes place in the plant cells. So I will say one point, that in plant cells furrow formation is absent. It does not take place. What is the reason? Because of the presence of what? Because of the presence of rigid cell wall. Because the cell wall which is present is rigid, that is why it will not allow the formation of what? Furrow. That is why furrow formation will not take place. In this case, what will happen? The cell wall will form. Cell plate formation will take place. And what is cell plate? Cell plate is basically it represents the middle lamella. Okay, so I will say it represents middle lamella. And it is the precursor for cell wall formation. Okay, so let me explain you the cell plate formation method. So suppose this is the cell wall, and this is the plasma membrane. And suppose these are the two daughter nuclei. So suppose this is one daughter nucleus, and this is another daughter nucleus. These are the daughter nucleus. And as I have told you that in plant cells, the interzonal spindle fiber, they will remain, they will persist. So suppose these are the interzonal spindle fibers. Suppose these are the interzonal spindle fibers. Okay, and these interzonal spindle fibers, they will form the phragmoplast. They will form the phragmoplast. Okay, so you can write that part over here. And in some books, it is written that the phragmoplast is formed by what? Phragmoplast is formed by Golgi body. But actually, the phragmoplast is nothing but the interzonal spindle fiber. You can write that part. Okay, now what will happen? Golgi body will secrete some vesicles. It will produce some vesicles, and those vesicles will come and they will lie at the center of the interzonal spindle fiber. So let me draw that. So suppose this is one vesicle, this is vesicle. So these are the vesicles, Golgian vesicles. Which vesicles? Golgi body Golgian vesicles. Okay, and they will have certain chemicals. They will have certain chemicals. Okay, so inside them, what is present? Some chemicals are present. Some chemicals are present, and these are basically pectate. These chemicals are basically pectate. Okay, now what is going to happen? So let me label them. So these are Golgian vesicles, and they have pectate. And what are these? These are the interzonal spindle fiber. These are interzonal spindle fiber. Now what will happen? These Golgian vesicles, they will fuse with each other. They will fuse with each other. So let me draw this diagram. So suppose this is nucleus, this is nucleus. Now what is going to happen? See, now I'm not going to mention the interzonal spindle fiber. Okay, they will be present, but there is no need to mention that again and again, so I'm not mentioning that. Okay, now what will happen when these suppose this is one vesicle and this is another vesicle. So when they fuse with each other, then what will happen? This membrane will fuse with this membrane, and this membrane will fuse with this membrane. Okay, so I will get a structure like this. So this is the membrane, and this is the membrane. These are the membranes, and these membranes are basically derived from what? Derived from these membranes of the Golgian vesicles. Okay, and the content which are present inside the vesicles, they will also fuse with each other. Okay, so they will also fuse with each other. So this is the structure. Okay, and this structure is called as cell plate, and these are the membranes. So let me label them. So what is this? This is membrane. Okay, and what is this? This is cell plate. Okay, now just a minute. Let me use some other color. These are the nucleus. Okay, now what will happen? Now more Golgian vesicles, they will come and they will fuse with the existing cell plate. And within them what will be present? Pectate will be there. Calcium pectate will be there. What will be there? Calcium pectate will be there. Okay, so these vesicles, these vesicles, they will fuse with the existing cell plate. Okay, and because of the fusion of the existing cell plate, what will happen? The growth of the cell plate will occur. Okay, so what will happen now? Suppose this is nucleus, this is one nucleus, this is another nucleus. Okay, so these are the nucleus, and now what is going to happen? So the chemicals present in this vesicle will fuse with this cell plate. Okay, so the cell plate will grow in size. So this has grown in size. Okay, and the membrane will also grow in size. So these are the membranes. So how the growth took place? See, the growth took place like this. So if this is, suppose this is cell plate. What happened? the chemicals got deposited over here, so it grew like this. It grew like this. That means the growth is taking place from center to outside, from center to outside. That is why I will say the growth of the growth of the cell plate is centrifugal. Okay, but the growth of furrow is centripetal. Okay, so here what is happening? Here centrifugal growth of cell plate. The cell plate grows centrifugally. The cell plate grows centrifugally. Okay, and finally like this happens. Okay, so here let me draw some other vesicles here. Okay, so see, suppose these are the vesicles, and these vesicles will again and again come and bind to the existing cell plate. Okay, and they will cause the growth of what? Cell plate. They will cause the growth of the cell plate. And finally what will happen? This membrane and this membrane, they will fuse. This membrane will fuse with this plasma membrane, and this membrane will fuse with this plasma membrane. This membrane will fuse with this plasma membrane, and this one will fuse with this plasma membrane. Okay, so they are going to fuse with plasma membrane. They are going to fuse with plasma membrane. So this is plasma membrane and this is plasma membrane. That means this membrane is ultimately going to form the plasma membrane, and this part will form the middle lamella. This part will form the middle lamella. Okay, so let me draw the middle lamella over here. Okay, so this is the middle lamella. This is the middle lamella. So see, and you can see inside this there will be nucleus. There will be nucleus. So this is nucleus. This is also nucleus. And what happened? Cytokinesis took place. So during cytokinesis, what is the structure which is formed first? It is the middle lamella. It is the middle lamella. So let me label them. Okay, so this structure is middle lamella. This structure is middle lamella. Okay, this structure is cell wall, and this structure is plasma membrane. This structure is plasma membrane. So during cytokinesis, the first structure which is formed is the middle lamella. Then later on, what will happen? Later on, so this is middle lamella, this is plasma membrane. Okay, so let me write here. So this is cell wall, this is cell wall, this is cell wall, and what is this? This is middle lamella. This is middle lamella. Now what will happen? This cell wall will be deposited. Which one will be deposited? Cell wall will be deposited. Okay, so let me deposit this cell wall. Okay, initially primary wall will be deposited. Later on, the secondary wall will be deposited. So let me draw the cell wall over here. So this cell wall gets deposited. Which one gets deposited? The cell wall gets deposited. Okay, so this is what happens. This is what happens. Okay, so I will say here what happened? Formation of middle lamella. So this is middle lamella. Later on, what happens? Formation of cell wall. So I can write here formation of cell wall. Formation of cell wall. In this case, what happened? Formation of middle lamella. And plasma membrane. Okay, in this case, what happened? The fusion of Golgi vesicles to form the cell plate. So I will say to form cell plate. Okay, so we are done with the mechanism of cytokinesis. Now we will talk about the significance of mitosis. We'll talk about significance of mitosis. Okay, let me check the time first. It is 2 hours and 38 minutes. Okay, no issues. Now, what about the significance of mitosis? Let me take some water and I will teach. So what are the significances of mitosis? Okay, so let me write here. So the first significance of mitosis is growth. Okay, second significance is repair. Third significance is restoration. Okay, fourth significance is also restoration. So restoration of what? Restoration of high surface area upon volume ratio of cell. Okay, and here high nucleocytoplasmic ratio. High nucleocytoplasmic ratio. Nucleocytoplasmic ratio, which is also called as karyocytoplasmic ratio. Which is also called as karyocytoplasmic ratio. Okay, the growth is basically increase in number of cells and increase in mass, and number of cells increases due to cell division. And remember one thing, the growth of organism is due to either mitosis or amitosis. So I will write here the growth, which cell division growth is because of due to increase in number of cells. Okay, growth is due to increase in number of cells, and the number of cells increases due to cell division. Okay, and so I will write here that the growth is due to increase in number of cells. Okay, and the number of cells increases due to cell division. So which cell divisions are responsible for growth? The cell division can be mitosis, it can be mitosis or amitosis. It can be mitosis or amitosis. Okay, so mitosis takes place in eukaryotic cells for growth, in eukaryotic organisms, and amitosis takes place in prokaryotic organisms for growth. Okay, so this was about growth. And remember one thing, in case of plants, the meristems which are present in specific locations, they undergo mitosis. For continuous growth. And that mitosis will cause continuous growth of plant. Okay, what about repair then? See, suppose some cells get damaged. Okay, and if some cells get damaged, then what will happen? This should be replaced, and that replacement is because of mitosis. Okay, for example, the cells of upper epidermis, they are continuously replaced, or the cells of lining of the gut, they continuously get repaired, or the cells of blood, they continuously get repaired. By what? By the mitosis. And remember one thing, in animals growth takes place up to a certain age, but cell division continues. We have learned that growth is due to cell division. And in animal growth takes place up to a certain age, but cell division continues. So if the cell division is continuing after a certain age, when the growth has stopped, then why the cell division takes place? To repair the damaged cells, or to replace the damaged cells. Okay, so I will say the repair of damaged cells. What are the examples? So I will say replacement of cells of what? Blood, or what? Cells of what? Blood or upper cells of epidermis, or cells lining gut. Okay, what about restoration of high surface area upon volume ratio and restoration of high nucleocytoplasmic ratio? Let me explain you with the help of one diagram. Okay, so suppose this is a cell. Okay, and this cell underwent mitosis to produce two daughter cells, and the daughter cells will be smaller in size. So this is one small daughter cell, and this is one daughter cell. And what will happen these two daughter cells, they will undergo growth, okay, interphase, and finally they will become large mature daughter cells, okay, and then these cells, they will go mitotic phase to produce the daughter cells, small daughter cells. They will form these small daughter cells, okay. So in this case, what is happening? Mitosis is taking place. What is taking place? Mitosis is taking place. So this is mitosis, okay, this is mitosis. Mitosis is taking place. This is growth, or I can say interphase, okay. These are the daughter cells and this is the parent cell. And what is this? This is mitosis, okay. This is large cell, this is small cell. Large cell will have, okay, let me write here, surface area upon volume ratio. Okay, see, for larger cells, the surface area upon volume ratio is small, whereas for smaller cell, the surface area upon volume ratio is large, okay. And when the surface area upon volume ratio is large, what will happen? There will be better exchange of materials, okay. It can easily uptake the nutrients from outside, and it can easily release the waste material from the cell. You can note that point. High surface area upon volume ratio, it allows better exchange of materials, uptake of nutrients and release of waste materials. You can write this point, okay. And again, this cell became large, so surface area upon volume ratio will become low, and here the surface area upon volume ratio is large. So see, when the daughter cells are formed, initially they had high surface area upon volume ratio there, and that is why they were able to uptake nutrients easily. But with growth, what happened? Surface area upon volume ratio decreased, and that is why now the uptake of nutrients and release of waste material will become slow. And to have higher and to achieve the higher rate of exchange of nutrients, uptake of nutrient and release of substrate, it has to again gain the condition of high surface area upon volume ratio. And to have high surface area upon volume ratio, this cell will undergo division to produce the small cells. Small cells, they have high surface area upon volume ratio. And how small cells are produced? Due to cell division, mitosis. See, initially the cell had high surface area upon volume ratio, okay, but because of growth, the high surface area upon volume ratio became decreased, low. And because of mitosis, what happened? Again, the high surface area upon volume ratio is achieved. So I can say, because of mitosis, what is happening? Because of mitosis, high surface area upon volume ratio is restored. Initially it was high, because of growth it became low, and because of mitosis, what happened? This ratio again become high. That means high surface area upon volume ratio was restored. Because of what? Because of the mitosis. Now here let me write nucleocytoplasmic ratio. For that I am writing N upon C. Okay, for smaller cells nucleocytoplasmic ratio will be large, and for larger cell nucleocytoplasmic ratio will be small, okay. And if this cell has high nucleocytoplasmic ratio, the control of nucleus, the control of the activity of cell by the nucleus will be easier, effective, efficient, okay. So in small cell, the control of the cell activity by the nucleus is better, but as the cell increased in size, the control, because of the decrease in the nucleocytoplasmic ratio, the control is less efficient, and that is why it has to become more efficient. And to achieve this, mitosis took place to restore the high nucleocytoplasmic ratio, okay. So you can write one point over here, what? High nucleocytoplasmic ratio allows better control of cellular activities by the nucleus, okay. So this was about significance of mitosis, okay. So then let's move ahead. So this is the diagram of various phases of Mitosis, you can clearly see that this is interphase, okay, in which chromatids are present and two daughter centrosomes are present. In prophase, the centrosomes are moving away from each other, and during that movement, astrole rays are being formed. And then in metaphase, what is happening? The metaphasic plate is formed, okay, and all the chromosomes, they lie at the single plane, okay, and then the sister chromatids, they get separated in the anaphase, and finally the telophase comes, in which the two daughter nuclei are formed, okay, and finally the cytokinesis will take place to produce the two daughter cells, okay, and both the daughter cells, they will undergo again interphase, and they will then the prophase and metaphase, like this, so the cycle will continue. And this is the diagram of NCERT. This is metaphase, this is transition to metaphase. In transition to metaphase, the spindle fibers, they go and get bind to the kinetochore of chromosomes, but the chromosomes are scattered. Whereas in metaphase, the chromosomes are aligned on a single plane to form the metaphasic plate. And one more thing, in this diagram you can clearly see that the smaller chromosomes, they are present on the outer side periphery, whereas the larger chromosomes, they are present at the center, okay. Now let's talk about the second type of division, that is meiosis, okay. And this meiosis consists of two phases, two subphases, meiosis first and meiosis second, okay. So suppose this is a cell, which is undergoing meiosis, and the cell which undergoes meiosis, that is said to be meiocyte. What do you call that cell? You call that cell as meiocyte. So this is meiocyte. This cell is called meiocyte, okay. Any cell which undergoes meiosis, that is meiocyte. Cell that undergoes mitosis is called as mitocyte, okay. So this cell will undergo meiosis first, then meiosis second, okay. So when this cell meiocyte undergoes meiosis first, it will produce two cells, and these two cells, they are called as dyad. Okay, and both the cells of the dyad, they will simultaneously undergo meiosis second. That means this one and this one, they will simultaneously undergo meiosis second to produce four cells, okay. And these four cells, they are collectively called as tetrad, okay. And these four cells are the daughter cells of meiosis. These two cells, the dyads, they are not the daughter cells. Important. This is meiosis first, okay. We said meiosis first and meiosis second, they are collectively called as meiosis. So we cannot call these dyads as product of meiosis. These four cells, the tetrads, are the products of meiosis, okay. And suppose the meiocyte is diploid in nature. This is diploid in nature. So diploid cell, we can write that as 2n, okay. And in the dyad, the chromosome will be, the number of chromosome will reduce to half, so it will, from 2n it will become n, okay. And in meiosis second, there will be no change in chromosome number, so that is why it will be n, it will be n, it will be n, and it will be n, okay. So one thing you can clearly see that in meiosis first, there is reduction in chromosome number from 2n n, okay. And that is why this meiosis first is considered as what? This is considered as reductional division, okay. Which one? Meiosis first is reductional division. Why reductional division? Because there is decrease in the chromosome number. From 2n it became n. But in meiosis second, the cell is n and this cell is also n. That means there is no change in chromosome number, and that is why this division is equational division. This division is equational division. Which one? Meiosis second is equational division, okay. And I said meiosis first and second collectively called as meiosis, and in meiosis what is happening? 2n cell is getting converted into n. So I can say meiosis is reductional division, okay. So meiosis is reductional division. Why? Because the meiocyte, which is 2n, gets converted into n, and that is why that means that the chromosome number is decreasing, hence we call this as reductional division. But both the phases of meiosis, they are not reductional. Only meiosis first is reduction, whereas meiosis second is equational. Important point, okay. And now in every sexually reproducing organism, what happens? Meiosis and fertilization takes place, okay. In every sexually reproducing organism, at some point of time, meiosis will take place and fertilization will also take place, okay. I'm not saying that every time the meiosis will take place to produce the gamete. No, It is not necessary that sexually reproducing organisms, they will produce gamete by meiosis only. No, by mitosis also gamete can be produced. Okay, but at some point of time of the life cycle, the sexually reproducing organisms, they will show meiosis. Okay, and when meiosis takes place, what happens? The haploid phase is achieved. Which phase? Haploid phase is achieved. Okay, and when fertilization takes place, what happens? The diploid phase is restored. The diploid phase is restored. Okay, so let me explain you this statement with the help of life cycle of plant. So in plant, we have two different types of bodies. One is sporophyte, which produces spore, and that sporophyte is diploid in nature. And that sporophyte will produce spore, and this spore is haploid in nature. Spore is a cell. So this cell, when this cell is produced, that means there must be some division. Okay, and what type of division will take place over here? It is the meiosis. So when meiosis took place, what happened? The 2n phase, that is diploid phase, produced the haploid phase, N phase. Okay, now what will happen? The spore will germinate to produce the gametophyte, and again the gametophyte is going to be N. Okay, and the gametophyte will produce gametes by the process of gametogenesis, and the gametes are N, and male gamete and female gamete, they will fuse with each other to form the zygote. Okay, and the zygote is 2n. Okay, and the process is fertilization. What do you call this process? You call this process as fertilization. Okay, so you can clearly see that when meiosis is taking place, what is happening? 2n is converted into n. That means haploid phase is produced. Okay, and whereas when fertilization is taking place, what is happening? The haploid phase is getting converted into diploid phase. Okay, and that is what I have mentioned over here. And here I have talked only with respect to what? Chromosome number. Why? Because when we define a division equational, we refer a division as equational or reductional, we consider only the number of chromosomes, not the amount of DNA. So what about the amount of DNA then? See, for amount of DNA, we use the symbol C. Okay, so suppose this cell has 2C amount of DNA. 2C amount of DNA. So here also the amount of DNA will be 2C. Okay, that means in meiosis first, what is happening? Only the chromosome number is getting decreased, but not the amount of DNA. Here the amount of DNA is 2C. Here also the amount of DNA is 2C. But in meiosis second, what is happening? Chromosome number is same, but the amount of DNA will get reduced. Okay, so from 2C it is getting C. Okay, so here it will be C. So you can remember this one. Okay, fine. And now what about the types of meiosis? Okay, I'm just writing this, I'm just saying this, you can note those points. Meiosis is basically of three types. What are those? One is gametic meiosis, second is sporic meiosis, and third one is the zygotic meiosis. So what do you mean by gametic meiosis? Any meiosis which produces gamete. That means the product of, if the product of the meiosis is gamete, then we call that meiosis as gametic meiosis. So in gamete mother cell, meiosis will take place to produce the gamete. And in which organisms do you find such type of meiosis? In organisms showing diplontic life cycle. So any organism which shows diplontic life cycle, that will show meiosis to produce the gamete, and that type of meiosis is called as gametic meiosis. And the examples of such organisms are animals, gymnosperms and angiosperm, and few algae like fucus. Okay, so you can note down those points. What? Gametic meiosis, meiosis that produces gamete. Observed, second point, observed in organisms showing diplontic life cycle. Example, fucus, which is an algae. Okay, and gymnosperm, angiosperm and animals. Second type of meiosis is sporic meiosis. If the product of meiosis is spore, that meiosis is called as sporic meiosis. Okay, if the product of meiosis is gamete, gametic meiosis. If it is spore, it is said to be gametic meiosis, so sporic meiosis. So in which organisms do we find sporic meiosis? In organisms showing haplodiplontic life cycle. For example, bryophyte and pteridophyte. Okay, so you can note down these points. Okay, and apart from that, few algae also show haplodiplontic life cycle. Okay, now like kelps. Okay, and what about zygotic meiosis? In zygotic meiosis, zygote is not the product of meiosis, but rather, I would say, rather, in zygote meiosis takes place. Okay, so meiosis which produces gamete is gametic, meiosis which produces spore, sporic meiosis, meiosis which takes place in the zygote, that is zygotic meiosis, and this takes place in organisms showing haplontic life cycle, for example most of the algae. Okay, so this was about types of meiosis. You can note down those points. Okay, now let's talk about some more point. So if I talk about mitosis, in mitosis we know there will be interphase which will consist of what, G1S and G2 phase, and then the karyokinesis which will include prophase, metaphase, anaphase and telophase, and finally the cytokinesis to produce the two daughter cells. Okay, so here I can say that these three phases they are collectively forming what, interphase. Okay, and these four phases they are collectively forming what, karyokinesis. Okay, and I can say that all these phases they collectively form mitotic phase. Which phase? Mitotic phase. So I can write here, this is interphase. Okay, so this is interphase. Okay, and this one is karyokinesis. This one is karyokinesis. Okay, and this entire process phase is mitotic phase, M phase. Okay, now we know that in S phase what is going to happen? In S phase, DNA will replicate. Which one will replicate? DNA replication will take place. And apart from that, in S phase, centrosome duplication also takes place. Okay, centrosome duplication takes place. Okay, and in anaphase what happens? In anaphase, I'm talking about anaphase only. In anaphase there is separation, and separation of what? Separation of sister chromatids. Separation of sister chromatids takes place. Okay, so in this case, in case of mitosis, I can say that in mitosis what is happening? One round of DNA replication is taking place, which is followed by one round of separation of chromatids, sister chromatids. So, and that is why I will say in mitosis, therefore we can say that in mitosis what is happening? I can say that one round of DNA replication is followed by what? I would say one round of separation. And who are going to get separated? I would say sister chromatids. Okay, and here what is happening? Karyokinesis is taking place and cytokinesis is taking place. That means division is taking place. So I will say one round of DNA replication is followed by one round of division, which will include division of nucleus as well as division of cytoplasm. So I will say followed by one round of what? Division. Okay, one round of division. So I will say one round of division. Okay, now if I talk about meiosis, then in meiosis also, before the M phase of meiosis, what happens? The interphase will take place. Which phase? Interphase will take place. Okay, so this is the interphase. So this one is the interphase. Okay, and I would say prophase, metaphase, anaphase, telophase first. Just a minute. These four phases they are collectively said to be what? Karyokinesis first. Okay, and these four phases they are collectively called as what? They are collectively called as what? Karyokinesis second. Okay, karyokinesis second, right? And I can also say that just a minute, This one is meiosis first, okay, and this one is meiosis second. This one is meiosis second, right? This one is meiosis second. So let me write that part. Okay, so this part is karyokinesis first. This part is karyokinesis first, okay, and this entire part is meiosis first, and this part is karyokinesis second, and this entire part is meiosis second. The entire part is meiosis second, okay. And in S phase, what is going to happen in S phase? The DNA will replicate, so I will write DNA replication takes place. And not only that, in S phase, the centrosome will also duplicate. Centrosome duplication takes place. That means whether the cell division is mitosis or meiosis, it does not matter, but in both cases, the interphase is the first phase which is going to take place. Okay, and that means before every division, that whether that is mitotic or meiotic, the DNA will replicate and centrosome will also duplicate if centrosome is present. Okay, and then in Anaphase first, what is going to happen? In Anaphase first, there is separation of what? Separation of homologous chromosome will take place. How the homologous chromosomes will separate, we will learn it later on, okay, in the same in this class only. Okay, so homologous chromosomes, they get separated. Which chromosomes? Homologous chromosomes, they get separated when? In the Anaphase first, and in Anaphase second, the sister chromatids, they get separated. Separation of sister chromatids. Sister chromatids take place. Okay, and what about interkinases, which is also known as intrameiotic interphase? In this case, there is no DNA replication, but there is duplication of centrosome. Okay, so I can write here that in this phase, what is going to happen? No DNA replication. There is no DNA replication, but there is centrosome duplication. So but centrosome duplication takes place. But centrosome duplication takes place. Okay, DNA replication does not take place, but centrosome duplication takes place. Okay, so here I can say that in meiosis, one round of DNA replication is followed by 2 rounds of separation. In Anaphase first, there is separation, and in Anaphase second also there is separation. Or I can also say that one round of DNA replication in meiosis is followed by 2 rounds of division. This is one division, this is second division. Okay, but in mitosis, one round of replication is followed by 1 round of division and 1 round of separation. Here, 1 round of DNA replication is followed by 2 rounds of separation and 2 rounds of division. Okay, and that is what I'm going to write here. Okay, I'm going to write here. So I will say in meiosis, one round of DNA replication, one round of DNA replication is followed by, what? 2 rounds of separation and 2 rounds of division. Two rounds of division. Okay, so I will say there are 2 rounds of divisions. Okay, so this was about a basic difference or comparison between mitosis and meiosis. Now let's talk about the meiocyte. What is meiocyte? Any cell which undergoes meiosis, that is called as meiocyte. So I will say cell undergoing meiosis, or the cell which undergoes meiosis, that is called as meiocyte. And the meiocyte is always even number ploidy. Okay, that means it can be, this can be diploid, can be tetraploid, can be hexaploid, etc. But it is never what? It is never 3n, I can say n haploid. It is never going to be haploid. The meiocyte is never going to be haploid, nor triploid, nor pentaploid, etc. That means the ploidy level of the myocyte will always be even, not odd one. Okay. Now, so how does this meiosis takes place? Let us learn that. So we will talk about prophase first. We are not going to learn about what interphase, because we are already done with the interphase. Okay. So the prophase first is, if I compare the prophase first with the prophase of mitosis or the prophase second of the meiosis second, then we will learn that this prophase first is more complex and this is more elaborated. Okay. Why? Because in this prophase first, a number of events are going to take place, and to explain those events, we have subdivided this prophase first into 5 subphases, and you will be asked about the sequence of these subphases. Okay. And what about the sequence of these subphases? The first subphase is leptotin, then zygotin, then diplotin, then pachytin, then diplotin, and finally the dikinases. Okay. In mitosis, the prophase has no subphases, or no subphases like this one. Yes, no doubt, the prophase of mitosis is divided into early prophase, middle prophase, and late prophase, but not like leptotin, zygotin, pachytin, diplotin, dikinases. And in prophase second of meiosis second also there is no subphases like this one. Okay, and that is why I said that this prophase first is more elaborated and more complicated or complex as compared to prophase of mitosis and prophase second of the meiosis second. Okay, now, so what are the events? So the first question which will be asked is the sequence of these subphases. The second question will be the second question which may be asked to you is the events which take place in the different subphases of prophase first. Okay. So in the leptotin, what is going to happen? See, the leptotin is the first subphase of what? Prophase first. And the prophase first occurs just after the interphase. And in interphase, we have the chromatins. So now the chromatins will start to condense. Okay. So the condensation of chromatin starts in which phase? Leptotin phase. But the condensation will complete by dikinases. That means by the end of prophase first only, only the chromatins will the condensation of chromatin or chromosome will complete. But in mitosis, the condensation of chromatin or the chromosome completes by metaphase. Here it starts in prophase first and ends by prophase first itself. Okay, so here the condensation of chromatin starts, okay, and that will produce the chromosome. And because the condensation has just started, that means the chromosome which is formed is not completely condensed. That is why the chromosome is going to be thinnest and longest. Okay, and here the chromatids are not visible. Okay, and one end of each chromosome will be facing towards the end, which will have the daughter centrosome. So let me explain that part. Okay, so suppose this is the plasma membrane, and suppose this is the nuclear envelope. This is the nuclear envelope. And now suppose these are the suppose these are the daughter centrosomes. These are the daughter centrosomes. Okay, these are daughter centrosomes. And now let me draw the chromosomes over here. Which one? Chromosomes over here. So suppose this is one chromosome. Okay, this is another chromosome. This is another chromosome. So these are the chromosomes. These are the chromosomes, and the end of chromosome is known as telomere. So you can clearly see that one end of every chromosome is converging towards the end which is having centrosome, daughter centrosome, and that is why if the appearance of these chromosomes is just like bouquet, and that is why we call this phase as bouquet stage. What do we call this stage? A bouquet stage. Okay, and these chromosomes, these chromosomes will have a number of swellings. Okay, so these swellings are called as chromomeres, and in these swellings the amount of DNA present will be more as compared to the other regions. Okay, so these swellings which are having more amount of DNA as compared to other regions, they are called as what? Chromomeres. So a number of chromomeres will be present. Okay, so this is how the structure will appear, the cell will appear in the leptotin phase. Okay, and one more thing that if this is the nuclear envelope, okay, and suppose this is the chromosome, okay, and these are the chromosomes, then what will happen? and these chromosomes are attached to the nuclear envelope. Okay, and they are attached not directly but through a structure. So this is a structure, and this structure is called as what? Attachment plate. This structure is called as attachment plate. So I will say the telomere or the end of chromosome is attached to the what? Attached to the attachment plate, okay, of the nuclear envelope. I can say that. Okay, so this is what I have written over here. Okay, and now, and what will happen now we have two daughter centrosomes, and among these two daughter centrosomes, one will start to move. One daughter chromosome centrosome will start to move. So suppose this one is moving in this exit. Okay, in mitosis, in early prophase, both the daughter centrosomes move, but in this case only one will move, and both will form the what? Astral rays. So here the astral rays will be formed. Which one? Astral rays will be formed. So suppose these are the astral rays. Okay, and because the astral rays are formed, so I will say the aster is formed. Which one is formed? Aster is formed. Okay, so this is the structure of a cell which can be observed in the leptotin phase. Which phase? Leptotin phase. Okay, so let me label, you can label this, or I'm just saying this. Okay, these pairings are chromomeres. Okay, and these are the daughter centrosomes, and these are the astral rays. These are the astral rays. Okay, now what about the second phase? In the second phase of the prophase, first is zygotin, and in this zygotin, what is going to happen? The homologous chromosomes, they will pair with each other. Okay, chromosome 1 they are homologous. Chromosome 2 and 2 they are homologous. Chromosome 3 they are homologous. Chromosome 5 homologous. Chromosome 9 homologous. Chromosome 20 homologous. Okay, so the homologous chromosomes, they will pair. 1 2 they are non-homologous. 3 4 non-homologous. 1 3 non-homologous. X and chromosome 3 they are non-homologous. So non-homologous chromosomes, they will not pair with each other, but the homologous chromosomes, they will pair with each other. Okay, why? Because they have to exchange their material between them. The homologous chromosome are supposed to exchange their material, and that process is called as what? Crossing over. Okay, and if the suppose this is one chromosome and this is another chromosome, suppose this is chromosome 5, this is chromosome 5, homologous chromosomes, and they are and suppose they have to exchange their materials segments between them. So if they're far away from each other, then how can they transfer their segments? This is not possible. That means if the chromosomes, homologous chromosomes, they are far away from each other, there will be no crossing over, and that is why they have to come close to each other, and that pairing of chromosome is known as synapsis, which will allow the exchange of chromosome. So I will ask you, if I ask you why this synapsis or pairing of chromosome has to take place, your answer should be because only pairing of chromosome will allow further crossing over. Without pairing of chromosome or pairing of chromosomes, no crossing over can take place, and that is why the synapsis or pairing of chromosome has to take place. Okay, so the synapsis takes place, and what is synapsis? This is basically pairing of homologous chromosomes. Which chromosomes? Homologous. This is very, very important. Okay, and which one is going to perform this pairing? A complex is going to perform this pairing, and that complex is known as synaptonemal complex. Okay, so whenever synapsis or synaptonemal complex terms are given, you have to think of what? Only zygotin phase. Okay, now when the two homologous chromosomes pair with each other, the paired chromosomes are now called as bivalent. What do you call that as? Bivalent. So when chromosome 1 and 1 pair, that will form one bivalent. 2 and 2 when they pair, that will form one bivalent. Okay, so bivalent will be formed. Okay, so suppose this is chromosome 1, this is chromosome 1, this is chromosome 2, this is chromosome 2, and this is chromosome 3, this is chromosome 3. So like this we have different chromosomes. So when these two will pair, that will form one bivalent. When these two will pair, they will form one bivalent. This two will pair, they will form one bivalent. And then when these two will pair, they will form one bivalent. And in this case, you can see every chromosome is present twice. So I can say this is a diploid cell. This is a diploid cell. And how many bivalents are there? 4 bivalents are formed. How many? 4 bivalents are formed. So I can say if the myocyte or the cell which is undergoing meiosis is 2n, then the number of bivalent will be n. Okay. So in our case, how many bivalents will be formed? 23 bivalents will be formed. 1 1 they will form one bivalent. 2 2 one bivalent. 3 3 one bivalent. Like this, 21 21 one bivalent. 22, 22, one bivalent. X and Y or XX, they will form one bivalent. So how many bivalents will be formed in case of human, whether that is male or female? 23 bivalents will be formed. So remember one thing, that number of bivalents will be n if the meiocyte is 2n. If the meiocyte is 4n, number of bivalent will be 2n. If the meiocyte is 6n, the number of bivalent will be 3n. Clear? So let me explain this point with the help of a diagram. And remember one thing, the condensation is still continuing, but the chromatids are not visible. The chromatids are not visible. Okay, so suppose this is one centrosome, okay, which is moving, and this is the other centrosome. Okay, and both of them, they are forming what? Astral rays. Both of them, they are forming the astral rays. Okay, and this is moving, okay, and we are having chromosomes. So suppose this is chromosome, suppose this is chromosome 1, okay, and this is chromosome 1. So these are the two chromosome 1, and suppose this is chromosome 2 and this is chromosome 2. These are the two chromosomes. Okay, and here I have to draw what? A nucleolus also. So suppose this is nucleolus. In this case, suppose this is the nucleolus. Okay, nucleolus will be there, and here also I have nucleolus. I will be having nucleolus. Okay, so I'm using different colors, no issues because of lack of time. Okay, so this is nucleolus, this is nucleolus. Okay, and this one is chromosome 1, this one is also chromosome 1, this is chromosome 2 and this is chromosome 2, and both of them, they will have centromere. Okay, right? So suppose this is the centromere, this is the centromere. Okay, this is the centromere and this is the centromere. Okay, and these two chromosomes, they have paired with each other. Okay, and there will be synaptonemal complex in between them. There will be synaptonemal complex in between them. So suppose this is the synaptonemal complex, and these two chromosomes, they are now called as bivalent. Bi means two chromosomes are present, that is why we call them as bivalent. If suppose this is chromosome 1 and this is chromosome 1, when they are present separately, I will call this as monovalent. I will call this one as monovalent. But if they come close to each other, okay, and they pair with each other, now this complex will be called as bivalent. Okay, so the two monovalents, they will come close to each other to form a pair, and that pair is now called as what? Bivalent. What do you call this as? Bivalent. Okay, so this is the cell which is observed under the microscope during the zygote phase. Okay, now what happens? Now the non-sister chromatids will exchange their segments. Okay, we are in bivalent. How many chromatids are there? So how many chromatids will be there? 4 chromatids will be there, right? Okay, let me explain this part. See, as I said, in leptotin phase, the condensation starts. But in leptotin phase, even in the zygote phase, the chromatids are not visible. But in pectitin phase, what will happen? The chromatids will become visible. The chromatids will become visible. Okay, and because the chromatids become visible, and that is why the bivalent will appear to have 4 chromatids. Okay, one chromosome will have 2 chromatids. So in bivalent, how many chromosomes are there? 2 chromosomes. If 2 chromosomes, that means 4 chromatids will be there. And because one bivalent has 4 chromosomes, that is why I will call this bivalent as, sorry, not 4 chromosomes, 4 chromatids. That is why I will call this bivalent as tetrad. Okay, why bivalent? Because 2 chromosomes are there. Why tetrad? Because 4 chromatids are there. Why? Because 4 chromatids are there. So the number of tetrad will be equals to number of bivalent. Okay, so in which phase the tetrads are visible? I will say pectitin phase. In which phase the bivalent first appears? So I will say it is the zygote phase. In pectitin phase also bivalent is observed, but first it appears in the zygote phase. Okay, tetrads, it appears in the pectitin phase. Okay, now so let me draw that diagram. So suppose this is the centrosome. Okay, and these are the astral rays. Okay, and suppose this is the centrosome and these are the astral rays. Okay, and then suppose this is moving like this. Suppose this is moving like this, and suppose I'm drawing the chromosome now. Okay, so suppose this is one chromatid, this is another chromatid. Okay, so this is one chromosome. Okay, and suppose this is another chromosome. Okay, and suppose this is one chromosome, this is another chromosome, and they have paired with each other. Okay, they have paired with each other. Okay, now what is going to happen? Now see, this is these are two chromatids, and these two chromatids are now called as sister chromatids. These two are also called as sister chromatids, but these two will be called as non-sister chromatids. Now what will happen? Segment will get exchanged between the non-sister chromatids of two homologous chromosomes. Okay, so and that process is called as crossing over. So when the crossing over takes place, the structure will look like this. Okay, so let me draw this one. So suppose this is chromosome 1. Okay, and suppose this is another chromosome 1. Suppose this is another chromosome number 1. Okay, so you can see there is exchange is going to take place. Okay, and suppose this is chromosome 2. Okay, and suppose this is chromosome 2, another chromosome 2, and here I will say at this point the crossing over is going to take place. At this point the crossing over is going to take place. Not only at 2 points, at several points the crossing over can take place. Okay, here I have drawn only 2 points. So I will say this is the point of crossing over. This is the point of what? Crossing over. Okay, and this entire complex is now called as what? This entire complex is now called as tetrad. Okay, this is bivalent as well as tetrad. Why tetrad? Because it has 4 chromatids. How many chromatids? 4 chromatids are there. Okay, and it will also show the what? Nucleolus. So it is the nucleolus. Okay, so in this phase what is happening? The crossing over is taking place. In this phase what is happening? The crossing over will take place. Tetrad becomes visible, crossing over takes place. So what is crossing over? This is basically exchange of segments of non-sister chromatids between homologous chromosomes. So remember one thing, the keywords here are what? Non-sister chromatids of which chromosome? Homologous chromosomes. So remember this one. Okay, and this exchange takes place with the help of a complex which is known as recombination nodule. Synaptoanimal complex will be there, and now the recombination nodule will appear, and what will this recombination nodule will perform? It will perform the crossing over, and this recombination nodule has recombinase enzyme complex which will have 2 different enzymes, endonuclease and ligases. Okay, and when the crossing over takes place, what will happen? There will be genetic recombination which will contribute towards the evolution. So let us try to understand actually what happens in the crossing over. What are these different points? Okay, let me explain this with more diagram. So suppose this is one chromosome, okay, and this is one chromatid, this is another chromatid, and these two chromatids are called as sister chromatids. Okay, and these two sister chromatids, they are held by a cohesin protein. So this one is basically cohesin protein. Okay, and now I can say that suppose this is chromosome 5. Suppose this is chromosome 5 and this is chromosome 5. Okay, so each chromosome, they are having 2 chromatids. Okay, 2 chromatids. Okay, these two are sister chromatids, these two are sister chromatids, but these two are non-sister chromatids. Okay, now what is going to happen? I'm going to perform crossing over. I'm going to perform the crossing over here. So how the crossing over will take place? Let me explain you. These two chromosomes are homologous chromosomes. These are homologous chromosomes. So for that, let me write here chromosome 5. This is chromosome 5 and this is also chromosome 5. So the process of crossing over takes place between the homologous chromosomes. So how will it take place? Let me tell you. In the first step, what will happen? We said in recombinase nodule or recombinase enzyme complex, we have endonuclease and ligase. So in the first step, the endonuclease will play its role. So what will it do? The endonuclease will cut these chromatids. Okay, so at this point, suppose at this point there is a cut, and at this point there is a cut, at this point there is a cut, and at this point there is a cut. So there is cutting of segments of what? Chromatids. Okay, and the first step, what is happening? There is cutting, and after cutting, this will appear like this, this will appear like this. Okay, so which one is doing this one? So I will say the endonuclease is doing this one. Which one? Endonuclease. So I will say in the first step, what is happening? There is cutting by whom? Endonuclease. By whom? Endonuclease. Okay, so at this point there is a cut, at this point there is a cut. Okay, if there is a cut at this point, and here also there will be a cut. okay, who is doing this one, endonuclease is doing this one, okay. And after that, what will happen, there will be exchange of these segments, okay. So this is a segment of what, chromatid. This is also a segment of what, chromatid. Which chromatids? These two chromatid segments, these two segments belong to non-sister chromatids. So what will happen, this segment will get transferred over here and this segment will get transferred over here, okay. That means mutual exchange is taking place between what, two homologous chromosomes and between non-sister chromatids, okay. So the mutual exchange took place. So here what happened, exchange took place. What happened, exchange took place. Exchange of what, segments of non-sister chromatid, that took place. And after exchange, what will happen, these two segments, they will get joined with each other. These two segments will also get joined with each other. These two will get joined and these two will also get joined, okay. And who is going to do this one, it is the ligase enzyme. It is the ligase enzyme. So I will say this, in the next step what is going to happen, the joining will take place. Joining of the segments by whom, the ligase. By whom, ligase, okay. So the ligase enzyme is going to perform the joining of what, segments. What, segments. Which segments? Segments of non-sister chromatids, okay. And finally I will get a structure like this. So suppose, to make you understand, let me write here, suppose this segment is a capital A, this segment is capital B, and this segment is capital D. I'm not writing capital C because capital C and small c difficult to distinguish, okay. So suppose this will also be capital A, this is capital B, and this is capital C, capital D. This is capital D. And suppose this one is small a, this one is small b, and this segment is small d. So this one also be small a, small b, and small d, okay. This one is small a, sorry, capital A, capital A, capital B, capital B, capital D, capital D. This one is small a, small b, small d, small a, small b, small d, like this, okay. So this one is small a, okay. This is capital B, this is small b, okay. This is small b, this is small d, and this is small d. And this one is capital D, okay. This is capital B, and this is capital A, and this is also capital A, okay. And here I will say this is capital A, okay. And this is capital B, small b, okay. And this one is capital D, okay. And this one is capital B, this is small a, this is small a, this is small b, okay. And this is small d, and this one is also small d. So initially what was the combination? The initial combination was capital A was present with capital B and capital D, okay. And small a was present with small a, small b, and small d. So, and these two are called as parental combination. These two are called as parental combination, parental type, okay. Suppose A, B, D, they are genes. In this case, what is happening? Capital A, capital B, capital D is present. Small a, small b, small d are present. That means parental types are there. But in addition to that, in addition to these two, capital A, small b, capital D is also present. Which one? Capital A, small b, capital D is also present. And small a, capital B, small d is also present. Small a, capital B, small d is also present, and they are called as non-parental type, or you can say recombinant type. Which type? Recombinant type, okay. And this process, and because in this process there is genetic recombination, and that is why we call this crossing over also as genetic recombination, okay. So this is recombinant type or non-parental type, and these two are called as parental type, okay. Parental type, fine. So this is how the crossing over takes place. This is how the crossing over takes place between whom? Between the homologous chromosome and between the non-sister chromatids, and in this case the segments will get exchanged between the non-sister chromatids of two homologous chromosomes, okay. Here I have drawn on I have mentioned only one, but between two homologous chromosomes at multiple sites the crossing over can take place, okay. Now let's talk about the next phase, and the next phase is diplotin phase, okay. In diplotin phase, what happens? Both the nucleolus, just a minute, whether I have drawn the nucleus over here. Yes. In the diplotin phase, what will happen? The nuclear envelope as well as the nucleus, they will start to disappear. Okay, so in this case, the size of the nucleus will be smaller because it is disappearing. So you can write here disappearing nucleus, and this is disappearing nuclear envelope. Okay, and suppose this is one centrosome and these are the astral rays. Okay, and this is another centrosome and these are the astral rays. Okay, now the because the crossing over has already taken place, and that is why the synapse chromosome or paired chromosome, they will start to get separated. Okay, they will start to get separated. Okay, but they will not how this separation will take place. See, the pairing or synosis occurred only to allow the crossing over, and as the crossing over has already taken place, and that is why the unpairing of chromosome will take place, and the pairing of chromosome was due to synaptopnemal complex. Okay, and now the synaptopnemal complex will start to degrade, but it will not degrade at a time, it will degrade gradually. Okay, and that is why I will say there is partial breakdown of what? Synaptopnemal complex. And when there is partial breakdown of synaptopnemal complex, what will happen? The homologous chromosomes or this bivalent, the homologous chromosomes, they will start to unpair. They will start to unpair, but at certain points they will still remain attached. Okay, and those points at which they are still attached, they are called as chiasmata. Chiasma is singular, chiasmata is plural. Okay, and chiasmata are those points at which the crossing over has actually taken place, and because of the presence of chiasmata, the chromosomes will appear as X shape. Okay, so you remember one thing, at which phase the X shape of chromosome appears? It is the diplotin phase. Why? Because of the chiasmata. Okay, I repeat, chiasmata are actually those points at which the chromosomes still remain connected, attached. Okay, actually, if to be more specific, I should say the non-sister chromatids. The non-sister chromatids, they still remain attached. Those points are called as chiasmata. Okay, and they represent those points which have at which the crossing over has actually taken place. Okay, and because of that, which type of shape is observed in chromosome? X type of shape is observed in chromosome. How? I will explain you. Okay, so let me write that. So suppose this is one chromosome. Okay, suppose this is one chromatid. Okay, this is another chromatid, and suppose this is another chromatid of homologous chromosome. Okay, and this is another chromatid. Okay, and suppose this is cohesin protein, this is cohesin protein. So suppose this one is chromosome 5 and this one is chromosome 5. Okay, and you can see crossing over is taking place. Okay, and here you can see this point. At this point you can see this shape is X shape. Okay, this shape is X shape. So and this point is called as what? This point is called as chiasmata. This point is called as chiasmata. Okay, so I can say because of the presence of chiasmata, the chromosomes appear as what? X shape. Which type of shape? X shape. Okay, now so let me draw the diagram then. Okay, so suppose this is one chromatid. This is one chromatid. Okay, and suppose this is another chromatid of the same chromosome. Okay, and because already the exchange has taken place, so let me draw those exchange part. Okay, and okay, so here this is, suppose this is chromosome 1, this is chromosome 1. Okay, you can clearly see in this diagram that some segments have been already exchanged. Okay, you can see in this case, if I draw this diagram in larger form, you can see, suppose this is chromosome, this is one chromatid, this is another chromatid. Okay, and suppose this is the segment of non-sister chromatid, this is the segment of non-sister chromatid. Okay, and here this is, just a minute. Okay, so you can clearly see that, in this chromatid we have this one, okay, and this one. And see, this is, suppose this is 1, this is 2, and this is 3. So 1 and 2 and 3, they are segments of what? Non-sister chromatids. Okay, so this is how the crossing over takes place. Okay, that means that this is the result after the crossing over. Okay, so this is tetrad, okay, and like this another chromosome, the chromosome 2 will also show the same condition. Okay, so suppose this is chromosome 2. And let me draw the chromosome second. Then let me draw the chromosome second. Just a minute. Okay, like this. So this is chromosome 2, and this is chromosome 2. This is chromosome 2, and this is chromosome 2. And in this case, this point, and this point, these points are chiasmata. This point and this point, they are the chiasmata. Okay, so let me write this point, write this one. So this is basically chiasma. Chiasma is plural, singular. Chiasma is singular. Okay, chiasma is singular. So this is what happens in the diplotin phase. Okay, in diplotin phase, desynapsis. The pairing of chromosome is known as what? Synapsis. So unpairing of chromosome is known as what? Desynapsis. Unpairing of chromosome is known as desynapsis. How does this take place? Because of the partial breakdown of what? Synaptotonal complex. And because there is partial breakdown of synaptotonal complex, not complete breakdown, that is why the homologous chromosomes or the non-sister chromatids, they still remain at contact at some points. Okay, and those contact points are called as chiasmata, and those chiasmata actually represent the site where the crossing over has taken place. Okay, now and one more point that in the diplotin phase, may it may last for longer time period as compared to other phases. Okay, and in the oocytes of some vertebrates, not all vertebrates, but in oocytes of some vertebrates, this phase, diplotin phase, can last for several months or years also. And such long or prolonged diplotin phase is known as dictyotin phase. Which phase? Dictyotin phase. And in some cases, the oocytes of the vertebrates, they show special type of chromosome known as Lamb-Brush chromosome, and those Lamb-Brush chromosome, they actually appear in this diplotin phase, and that is why we call Lamb-Brush chromosome as diplotin bivalent, because one Lamb-Brush chromosome consists of two homologous chromosomes. Okay, and two homologous chromosomes, they form one bivalent, and that is why we call this Lamb-Brush chromosome bivalent. And because they appear in the diplotin phase, that is why we call Lamb-Brush chromosome as diplotin bivalent. Okay, diplotin bivalent. Okay, so in this phase, what is going to happen? Nucleolus, nuclear membrane, they start to disappear. Okay, and they will completely get disappeared in the dikinesis phase. Okay, and diplotin phase in the oocytes of some vertebrates, they can last for several months or years, and such long diplotin phase is called as what? Is called as dictyotin phase. This is important, and this is the phase in which the Lamb-Brush chromosomes, they appear. This is the phase in which the Lamb-Brush chromosome appear. Okay, now the last subphase of what? Prophase first, and the last subphase is the dikinesis. So in dikinesis, what is going to happen? Nucleolus and nuclear membrane, which had started to dissipate in the diplotin phase, they will dissipate completely in this phase. Okay, and one more thing, and the chiasmata will shift towards the telomere. Okay, so suppose this is the chiasmata. Okay, so this chiasma, and this is the telomere. Okay, this is telomere. So now what is going to happen? This chiasmata will shift towards the telomere. That means after some time, the diagram will appear like this. Diagram will appear like this. And so the chiasmata has shifted, and after some time the chiasmata, let me draw this one. Okay, so suppose this is the chiasmata. Okay, so after some time what will happen? The chiasmata has shifted. Its position, and after some time what will happen? The chiasmata will shift further like this. So you can see the chiasmata is shifting its position, and finally what will happen? Both of them, they will get completely separated. Okay, both of them, they will get completely separated. Why? Because the chiasmata is shifting its position, and this shifting of the chiasmata towards the telomere is known as terminalization of chiasmata. What is this? Terminalization of chiasmata. So terminalization of chiasmata takes place in the dikinesis. So remember this one. This is very, very important point. Which event is going to take place? It is the terminalization of chiasmata. Which one? Terminalization of chiasmata will take place. Okay, and at this point, what will happen? The two daughter centrosomes, they will lie at different poles. Okay, opposite poles. Okay, suppose this is one centrosome, this is another centrosome, and they have, and this is astral rays, this is astral rays. Okay, and now let me draw the centrosome. Okay, so this is moving like this, and in the dikinesis, this has rest at just opposite to this one. Okay, and the nucleate membrane and nucleolus, they will disappear completely. Okay, so now let me draw the chromosomes then. Okay, so suppose this is one chromosome. Suppose this is another chromosome. Like this. Okay, so suppose this is chromosome 1, this is chromosome 1, this is chromosome 2, this is chromosome 2, and here we have centrosomes. Sorry, here we have centrosomes, right? Here we have centrosomes. Okay, and they have produced, and now what will happen? Now they will start to produce the spindle fiber. So spindle fiber formation start in which phase? Dikinesis. And when dikinesis completes, that means I will say prophase first completes, and because after prophase, the prophase first, the metaphase first is going to start, and that is why we say that dikinesis marks the transition to metaphase first. Inside it says metaphase, but it is actually metaphase first. Okay, dikinesis is the last subphase of prophase first. So when dikinesis completes, that means the prophase first completes, and when the prophase first completes, the metaphase first will begin, and that is why we say dikinesis marks the transition to metaphase first. Okay, so the main key points which you have to remember is in leptotin, you have to remember bouquet stage. Okay, condensation starts. In zygotin, synaptonormal complex synapses. Okay, and bivalent will be formed. In pectitin, crossing over will take place. Okay, recombination nodule will come into play. Okay, and there is exchange of segments of non-cystic chromatids. In diplotin, chiasmata, you have to remember. X shape of chromosomes, you have to remember. Desynapsis, you have to remember. In dikinesis, you have to remember about terminalization of chiasmata. Okay, so these are the keywords you have to remember about those subphases. Okay, that will be completely enough for you. Now in metaphase first, what is going to happen? In metaphase first, the spindle fibers, they will go and bind to the chromosomes. Okay, and they will bring them towards the equatorial plate. But in this case, two metaphasic plates will be formed. How many? Two metaphasic plates will be formed. So let me draw that diagram over here. So suppose this is centrosome, and suppose this is centrosome, and suppose these are the astral rays. Okay, and let me draw the chromosomes over here. Okay, so suppose this is one chromosome. Okay, and suppose this is another chromosome. It will be having the segment of other chromosome also, right? So this is the other segment. Like this. right? So here I will have the segment of other chromosome. Okay, so suppose this is chromosome 1, this is chromosome 2, this is chromosome 1, and this is chromosome 2. Okay, and in this case what will happen, the spindle fibers, they will go and bind to the chromosomes. Okay, so suppose this is the spindle fiber, these are the spindle fibers, these are the spindle fibers, these are the spindle fibers, and some spindle fibers, they will develop and they will go to the opposite pole, and some spindle fibers, they will develop but they will not get bind to the any chromosomes. Okay, so in this case, suppose just a minute, suppose this is centromere, this is centromere. You can see the centromeres are lying at their single plane, and this is centromere, this is centromere, they are also lying at a single plane. So this will form one metaphasic plate and this will form another metaphasic plate. So how many metaphasic plates are there? Two metaphasic plates are there. How many? Two. So I will say this is one metaphasic plate. Sorry. This one forms one metaphasic plate and this one forms another metaphasic plate. Okay, so how many metaphasic plates are there? Two metaphasic plates are there. So I will say there are two metaphasic plates. These are the metaphasic plate, and in each metaphasic plate you can see there is one copy of every chromosome. Okay, one copy of chromosome 1, one copy of chromosome 2. Okay, so these are the metaphasic plates. So how many metaphasic plates are formed? Two metaphasic plates are formed. This is important. Just like mitosis, here also metaphasic plate or equatorial plate will be formed, but in mitosis only one metaphasic plate is formed. In this case two metaphasic plates will be formed. In mitosis one chromosome is attached by this spindle fiber coming from two poles, but in this case one chromosome is bound with the spindle fiber coming from only one pole, coming from only one pole. Well, in the anaphase, what is going to happen? These homologous chromosomes, they will get separated. These homologous chromosomes, they will get separated. Okay, so in this case what is going to happen? The homologous chromosomes, they will get separated, and because the homologous chromosomes will separate, and that is why there will be no splitting of these centromeres. Centromere will remain intact. Okay, if I want to separate the sister chromatids, I have to split this centromere, but because in this case I'm not separating these sister chromatids, I'm simply separating the homologous chromosomes. That is why there is no need to split these sister chromatids, split the centromere. So the centromere will remain intact. Okay, it will remain intact. Okay, so let me draw that diagram then. Okay, so suppose this is centrosome, this is centrosome, and suppose these are the astral rays, these are the astral rays. Okay, and now let me draw the chromosomes over here then. Okay, they will have the segment of other chromosomes also, right? Like this. Okay, and suppose this is the centromere. Okay, and here, and these are the spindle fibers. These are the spindle fibers, and in between them also there will be more spindle fibers will be formed, and those spindle fibers are called as what? Interzonal spindle fibers. With spindle fibers, interzonal spindle fibers. So here, if this is chromosome 1, this will be chromosome 1. If this is chromosome 2, this will be chromosome 2. So which one are getting separated? Chromosome 2 separating. That means homologous chromosomes are separating. 1 they are separating. That means homologous chromosomes are separating. Okay, so this was about Anaphase first. This was about Anaphase first. Okay, and after Anaphase first, the telophase first will take place. So by the end of Nfs first, the homologous chromosome pair will group of homologous chromosomes, they will reach to their respective poles. Okay, and once they reach their respective poles, what will happen? The nuclear membrane or envelope will reappear around them, and nucleolus will also reappear, and the chromosomes, they will undergo slight decondensation. Slight, I did not say complete decondensation, but I said slight decondensation, and that slight decondensation will not form the chromatin. Why? Because we know when chromatin undergoes condensation, it produces chromosome, and when the chromosome undergoes decondensation, it will produce chromatin. And in this case, chromosome undergoes decondensation, but slightly decondensation will take place, and that is why chromatin will not be formed. The chromatin will not be formed. Okay, so that is what I have written. Nucleolus, nuclear membrane will reappear. Okay, and when the nucleolus and nuclear envelope reappear, daughter nuclei will be formed. Two daughter nuclei will be formed, and there is slight decondensation. Okay, chromatins are not formed. Okay, and in this phase, what will happen? The spindle fibers and the astral rays, they will disappear. The spindle fiber and astral rays, they will disappear. So suppose this is centrosome, okay, and they are not associated with any astral rays anymore, and they are not associated with any spindle fiber anymore, because they have disappeared completely. Okay, and suppose this is nuclear envelope. Okay, so nuclear envelope, and suppose this is nucleolus. Okay, suppose this is nucleolus, so they reappear. Okay, and now let me draw the diagram of what chromosomes. So suppose this is one chromosome. Okay, and suppose this is another chromosome. They are having segment of other chromosome also. Okay, and suppose this is chromosome 1 and this is chromosome 2. Okay, like this. Here also I can draw. Okay, so this is okay. So in this case, if this is chromosome 1, this will be chromosome 2. This will be chromosome 2. Okay, so I can say this because the number of copies of each chromosome is 1, so I can say write this nucleus as n. And in this case, you can see how many DNA molecules are present in one chromosome 2. Here also two DNA molecules are present, so I will say this can be represented n upon 2c, and this can be written as n upon 2c. Okay, and what are these? These are the daughter nuclei. Okay, now what will happen? Now this cytokinesis first will take place, and when the cytokinesis first will take place, what will happen? This will divide. Okay, and this will be one daughter cell. This will be another daughter cell. Okay, so like this is going to happen. Okay, so I will say after telophase first, what will happen? The cytokinesis first will take place. What will take place? Cytokinesis first will take place. So I will say cytokinesis first takes place. Okay, so I will say that if this cytokinesis first takes place, what will happen? It will produce two daughter cells. It produces two daughter cells. It produces two cells. Okay, and those two cells are called as what? Dyad. Those two cells are called as dyad. Okay, and what about the number of chromosomes in them? It will be n. Okay, and what will be the amount of DNA? It will be 2c. Okay, so this was about after cytokinesis first. Okay, so I can write like this, I can draw like this. Okay, so if suppose this is one daughter cell, this is another daughter cell. Okay, and suppose this is the nucleus. Okay, and suppose these are the nucleolus. Okay, and let me draw the chromosome over here then. Okay, so suppose this is one chromosome. Okay, and suppose let me draw over here another chromosome. Like this. Okay, and these two are called as what? Dyad. These two cells are called as dyad. Okay, and this is what you are going to get after cytokinesis first. And after cytokinesis first, see what is this? This is centrosome and this is centrosome. And for separating the chromosomes or sister chromatids, I need two centrosomes. And that is why and because both of these cells, they have to undergo meiosis second, and that requires another centrosome. That is why I have to produce another centrosome. Okay, and this takes place in the interkinases. So in interkinases, what happens? The centrosome duplication takes place, but DNA replication will not take place. DNA replication is absent, whereas centrosome duplication takes place. Okay, and this is the phase between meiosis first and meiosis second. Okay, and in this phase there will be a slight decondensation, but still there is no formation of chromatin. Okay, there is no formation of chromatin. That means the decondensation takes place in telophase first as well as interkinases, but in both of these phases chromatins are not formed. Why? Because there is slight decondensation, not much decondensation. Okay, now what will happen after interkinases? There will be meiosis second. You may ask me, sir, why there is no DNA replication? See, the purpose of DNA replication is to make the amount of DNA double, so that after separation the number of DNA molecule will remain same. See, when I'm performing cell division, suppose mitosis, so first of all I'm doubling the chromosome now, the amount of DNA by DNA replication, so that when there is separation and there is formation of new cells, they will have same number of DNA molecules. Okay, so to produce the cells of same amount of DNA molecules, I performed DNA replication earlier. Before separating, I performed DNA replication. But in meiosis, what is our purpose? Our purpose is to reduce the chromosome number to half, or amount of DNA to half. So if I perform DNA replication in this phase also, then what will happen? Amount of DNA will become double. Okay, and if the amount of DNA will become double, then what will happen? The daughter cells formed after meiosis will have the same number of DNA and same number of chromosome as compared to the mother cell, myocyte. And that is not our purpose, and that is why there is no DNA replication. Because if DNA replication will take place, what will happen? There will be the daughter cells will also have the same number of chromosome, and because I do not have to, I don't want that. That is why there will be no DNA replication. Okay, now the meiosis second is quite similar to mitosis, but there are some differences. Okay, so the phases of meiosis second, they will show some differences with mitosis and some differences with the meiosis first. So those differences, dissimilar points I have mentioned over here. So in prophase second, what will happen? This slightly decondensed chromosome will undergo condensation. In prophase first, chromatins undergo condensation. In prophase of mitosis, chromatins undergo condensation. In both cases, in prophase of mitosis and prophase first of meiosis, what is happening? The condensation of chromatin takes place, but in this case, the condensation of what is taking place? Slightly decondensed chromosome is taking place. Okay, so this is one difference. Otherwise, there will be disappearance of nuclear envelope, disappearance of nucleolus. Okay, there will be formation of astral rays, etc., etc. Okay, in metaphase second, what is going to happen? Only one metaphasic plate will be formed. In mitosis also only one metaphasic plate is formed, but in meiosis first, metaphase first, what is happening? Two metaphasic plates are formed. Okay, so you can see here in metaphase first, how many metaphasic plates are formed? Two metaphasic plates are formed. But in, you can see in metaphase second, how many metaphasic plate is formed? One metaphasic plate is formed. That means the number of metaphasic plate formed depends on the metaphase. Okay, nature of metaphase. If the metaphase belongs to mitosis, 1. If it belongs to meiosis second, it will be 1. But if it belongs to meiosis first, it will be 2. Okay, in anaphase second, what is going to happen? The sister chromatids will get separated. And this is what happens in mitosis also. Okay, and because I have to separate the sister chromatids, that is why I have to split the centromere into 2. Okay, so in this case, the centromere splits into 2, whereas in case of anaphase first, the homologous chromosomes they separate. the homologous chromosomes, they separate and the centromeres remain intact, that does not split. Okay, but in this case what is happening, in this case I will say, in this case what is happening, these sister chromatids, they get separated. Okay, not homologous chromosome, and the centromere splits into two. Okay, and both the daughter chromosomes, they will have one centromere. And in telophase first, telophase second, what will happen? Two haploid nuclei will be formed in each cell. Okay, and after that, the cytokinesis second will take place, and after cytokinesis second, I will get the 4 daughter cells, and those 4 daughter cells are called as tetrads. They are called as tetrads. Okay, so this was about the mechanism of meiosis. Now let us talk about, so here you can see the diagram. Here you can see the diagram. Okay, so this is Reproduction phase, and this is the nucleolus. Okay, and here you can see, here you can see the centromeres. The telomeres, they have converged towards the end, which are having the centrioles or centrosomes. Okay, in zygote there is pairing of homologous chromosome. This is one chromosome, this is another chromosome. Okay, if this is chromosome 1, this is chromosome 1, chromosome 2, chromosome 2, these are called as bivalent. This is called as bivalent. Okay, and in this case you can clearly see there is these chromosomes. In these chromosomes, chromatids are not visible. Okay, and these are the astral rays, these are the astral rays. Okay, and in this diagram astral rays are not formed, but actually in leptotin phase also, leptotin phase itself, the astral rays are formed. Okay, and in Pachydian phase, what is happening? The crossing over is taking place. Okay, so these are the synapse chromosome or bivalent. Okay, and at this point what is happening? The crossing over is taking place. At this point the crossing over is taking place. Okay, and after that what is happening? They will get separated, but they are still attached at this point, and these points are called as chiasmata. Okay, they are chiasmata, and in diakinesis they will get completely separated. In this diagram they are not completely separated. Okay, but they will get completely separated. Okay, so this was about what? This was about the various phases of what? Meiosis. Okay, and this is the diagram of what? NCERT. You can see this. Okay, prophase first, okay, metaphase first, anaphase first, telophase first. Okay, and after that you'll get the 2 daughter cells. Okay, and in these 2 daughter cells there will be prophase second, then metaphase second, anaphase second, and I will get the 4 cells, 4 nuclei at the end of what? Telophase second, or I can say 4 cells after the cytokinesis second. Okay, now what about the significance of meiosis? What about the significance of meiosis? Just a minute. I'm left with only 2 portions. Okay, let me check the time first. 1 hour. Okay, fine. 18 minutes. So the meiosis has 2 main significances. One is gametogenesis, second is maintenance of chromosome number generation after generation. So what is gametogenesis? Gametogenesis is the process of gamete formation. Okay, so I will say in many organisms, meiosis produces gamete. I did not say in all organisms, I said many organisms. Okay, so produces gametes. Okay, so let me give you one example then. So if I talk about bryophyte or algae, okay, or I will say most of algae, not all algae. So I will say most of algae. These are different organisms, and in these organisms, it is the mitosis, it is the mitotic division which produces gamete. And that is why I cannot say that it is always the meiosis which will produce gametes. No, normally it is the meiosis. Otherwise, in some organisms, mitosis also produces gametes. Okay, and that type of mitosis which produces gamete is called as gametic mitosis. Meiosis which produces gamete, gametic meiosis. Meiosis which produces mitosis which produces gamete, that is called as gametic mitosis. Okay, so this was one significance of what? Meiosis, that is to produce the gamete. Okay, so what is gametogenesis? This is the production or formation of gamete. The process of formation of gamete is called as what? Gametogenesis. And in some organisms, gametes are also formed by mitosis, which is called as gametic mitosis. In many organisms, the gametes are formed by meiosis, which is called as gametic meiosis. Now, the second significance, the most important significance, is the maintenance of chromosome number generation after generation. Okay. So, and this is called as what? This is paradox of meiosis. Okay. Why? Because if we see meiosis, actually meiosis reduces the chromosome number to half. Okay, and that is why it appears as if the meiosis is reducing the chromosome number, not maintaining the chromosome number. But if we try to understand it in deeper way, then we will realize that it is actually the meiosis which is maintaining the chromosome number. How? Let me explain you with the help of an example. So suppose this is one individual, this is another individual. Okay, suppose this has 2n number of chromosome, this has 2n number of chromosome. For example, this has 46 chromosome, this has 46 chromosome. Okay, so what I do is I perform meiosis to produce the gamete. So suppose this is gamete, this is gamete. Okay, suppose this is male gamete, this is female gamete. Okay, so here it will have n number of chromosome, it will have n number of chromosome. Which division did I perform? Meiosis. And what are these? These are the gametes. These are the gametes. Okay, this one is male and this one is female. Okay, and here I will have 23 chromosomes, here I will have 23 chromosomes. Okay, and when they fuse with each other, they will form a zygote. Okay, and this zygote will form the new individual, and it will have 22n chromosome number. That means 46 chromosome will be there. Okay, so you can see in this generation how many chromosomes are there? 46 chromosome are there. This is parental generation, and this one is offspring. And in offspring, how many chromosomes are there? 46. That means 2n chromosomes are there. Okay, and how this so in this parental generation, number of chromosomes is 2n, 46. Here in the F1 generation also it is 2n, 46. That means because of meiosis, the number of chromosome in the next generation is same. Okay, so let me explain you little more. Okay, so suppose this is one parent, this is another parent. Suppose this is male and suppose this is female, and both of them, they have 46 chromosomes. And suppose I performed mitosis to produce the gamete. So suppose this is gamete, this is gamete. So because I'm performing mitosis, so the number of chromosome will remain same, 46. Okay, and if I allow the fertilization, then I will get this zygote, and in the zygote there will be 92 chromosome. So this is parental generation, this is F1 generation. So if in this case I do not perform meiosis, then what is happening? The number of chromosome is doubling, not doubling. Okay, and that is why I will say the meiosis is the process which is actually responsible for maintaining the chromosome number same generation after generation. If there is no meiosis, and if there is no meiosis but fertilization is taking place, then what will happen? The number of chromosome will become double and double and double every time. Okay, so see, in sexual reproduction, what happens? Fertilization will take place. In fertilization, there is fusion of two cells, two gametes, which will have chromosomes, right? So before fusing these two gametes, actually what I do is I reduce the chromosome number to half, so that even after fertilization the number of chromosome will remain same. Okay, and that is why I will say it is the meiosis which is actually responsible for maintaining the chromosome number to chromosome number same generation after generation. But if we superficially see, then we will find that okay, meiosis is actually reducing the chromosome number, but actually it is maintaining the chromosome number generation after generation, and that is called as paradox of meiosis. Okay, and the another statement the NCERT says that meiosis itself, meiosis per se means by itself, reduces chromosome number to half, reduces chromosome number to half. Okay, now there are some formula you can see here. Okay, some formula are there, and remember one thing. So if you want to produce X number of cells, then how many meiosis do you require? You require X minus 1 mitosis. Sorry, if you want to produce X number of cells, by mitosis from a single cell, if you want to produce x number of cells by mitosis, then how many mitosis do you require? X minus 1. So if I ask you how many mitotic divisions are required to produce 16 cells, your answer should be 16 minus 1, that means 15. Okay. Now if you want to produce x number of cells from y cells after n number of mitotic generation, n number of mitotic generation, so what will be the value of mitotic generation? So I will say you want to produce x number of cells. Okay, so in this case x will be equals to what? Y into 2 to the power n. Y into 2 to the power n. So this is the formula you have to remember. Okay, so that means if you are initially provided with 10 to the power 5 cells, okay, and after 5 generation, or after 2 generations, how many cells will you'll get? So I will say 2 to the power 2 into 10 to the power 5. So this many cells you are going to get. Okay, now what about the number of meiosis to produce x number of these structures? So if you want to produce x number of microspores, or x number of pollen grains, or x number of zygote forming male gametes in angiosperms, then how many meiosis do you require? You require x upon 4 meiosis. That means if I want to produce 16 microspores, how many meiosis do I require? 4 meiosis I require. If I want to produce 16 pollen grain, I require 4 microspore. If I want to produce 16 male gametes in angiosperm, which will form the zygote, I require 16 upon 4, that means 4 meiosis. Okay, what about x number? What about the number of meiosis required to produce x number of megaspore, or x number of embryo sac, or x number of egg? It is x. That means if I want to produce 16 egg in angiosperm, I require 16 meiosis. If I want to produce 16 embryo sac, I require 16 meiosis in angiosperm. Okay, so what about the male gametes? See, we know, suppose this is 1 microspore mother cell. When it undergoes meiosis, it produces 1 microspore mother cell will produce 4 microspores. Okay, and all those 4 microspores, they will produce pollen grains, right? And each pollen grain has 2 male gametes. Each pollen grain has 2 male gametes. 2 male gametes are there, and between these two, out of these 2 male gametes, one will fuse with egg cell to produce the zygote. Okay, so I can say 1 meiosis is producing how many male gametes? 2 into 4 into 2, that means 8 male gametes are produced, and out of those 8 male gametes, only 4 will fuse with egg cells to produce the zygote. Okay, so I will say 1 meiosis is producing how many male gamete? 4 into 2, 8 male gametes are produced. How many zygote forming male gametes are produced? So I will say 4 zygote forming male gametes are produced. Why? Because in every pollen grain there are 2 male gametes, and out of those 2 male gametes, only one fuses with egg to form the zygote. Not both of them will fuse with the egg cell to form the zygote. Okay, so I will say to produce the x number of male gamete, I will require x upon 8 meiosis. So if I say if you want to produce 16 male gametes in angiosperm, how many meiosis do you require? I will say x upon 8. That means 2 meiosis you will require. What about seeds? Seeds are also called as grains or zygote or embryo. So if you want to produce x number of seeds, or x number of zygote, or x number of embryo, how many meiosis you require? You require x plus x upon 4. Okay, because to produce one zygote, I require what? I require 1 egg plus 1 male gamete, right? So if I want to produce x number of zygote, I require x male gamete plus x zygote forming male gamete and x egg cell. So here, as we know that to produce x number of egg, how many meiosis I require? X. So this x is actually for what? Egg, x number of egg. This is for eggs. So if I want to produce x number of egg, I require x meiosis, and this x upon 4 is for zygote forming male gametes. This is for zygote forming male gametes. So if I ask you how many meiotic divisions you require to produce 200 or suppose 100 seeds. So your answer should be 100 plus 100 upon 4. That means it will be 125. It will be 125. Okay. So here is a question. You can see somatic cell has just completed S phase of its cell cycle as compared to gamete. How many of the same species? See, somatic cell has just completed S phase. So the somatic cell which has just come, see, if the cell is somatic, that means that will be diploid in nature. That will be diploid in nature. So I will write 2n. Okay, and in somatic cell in G1 phase, it will be 2n upon 2c. Okay, and if it has just completed S phase, if it has just completed S phase, then the number of chromosome will remain same, but the amount of DNA will become 4 times. So double. Okay, so it will become 4c. Okay, and here it is talking about what? Gamete. It is talking about what? Gamete. The somatic cell that has just completed S phase of its cell cycle as compared to gamete. Okay, so in gamete, the gamete is haploid in nature. So let me write about gamete. So if I talk about gamete, so gamete is haploid in nature. So the number of chromosome will be represented as n, and the amount of DNA will be represented as c. Okay, so I can say that with respect to gamete, the amount of DNA is 4 times. Here c, here 4c, that means 4 times. And number of chromosome is, amount of DNA is 4 times. Okay, amount of DNA is 4 times, and number of chromosome is 2 times. Okay, so you can twice the number of chromosome and twice the amount of DNA. Not twice. Okay, 4 times the number of chromosome, not 4 times, 2 times. Twice the number of chromosome and 4 times the amount of DNA. So the answer is A. Okay, so this was all about the cell division. Okay, so hope it was helpful to you, and I will give you some questions. Okay, and you can solve those questions. Okay, and these questions you can find this PDF you can find in the PW app. Okay, so thank you so much, everyone. We will meet in the next class. And one student had asked me, sir, please teach plant physiology, and we are facing problem in that. No worries, we are going to teach plant physiology as well. Okay, so take care, everyone. Thank you so much. Bye.